tight. We'll be with you. Hopefully you can see both rooms at EBI at Broad, and we'll go through all the settings and getting everyone oriented momentarily. Let me share my screen. All right, is everything up on my screen?
Yes.
Very good. Give it another couple, just 1 minute till we start. How are you guys doing at EBI? Are you guys ready to go?
Yeah.
All right. Broad team, are you ready to go?
We are.
Excellent. See lots of people starting to join. A little bit less than yesterday, but we know how that goes. Hopefully people will join along the way. It's early for many people on the East Coast, so that's all right. We'll give them a little time to join and then get settled. Well, I want to welcome everyone back for day 2 of our community workshop on standards and infrastructure for predicted effector gene lists. And I'm really happy you're all back, and hopefully more people will join as we go, because today is really the important day of the 2, frankly, in terms of like actually synthesizing what we heard yesterday and we learned yesterday and trying to convert that into standards. And that will be a little bit challenging, right? Because we'll all have to have opinions, but the ultimate goal is to put those into a product of a more formulated straw man that we could share with the community. So just reminders, so everyone knows what things are. This is Zoom, not a webinar. We want this to be interactive, especially because we're going to be going to breakout sessions later in the day. We're going to have some whole group discussions over the course of the day and some breakout rooms. So hopefully you hang in there for the next 4 hours. I think it'll be all worth it. We're going to record these, not obviously the breakout sessions. I'm not sure how that would go, but we'll do the main sessions. Chat is open and active. I think it worked really well yesterday to capture questions. The questions from yesterday we've captured and kind of folded into our discussions today. So that's all there for you. There is an updated program. Go to that same Google Drive you got from Maria and Laura yesterday. It has all the materials, the program, our timing. We're going to try and keep to time. We added a little bit of intro from me, so hopefully that's okay. I think that we'll adjust over time. Our MCs will be me, Laura, and Maria, with technical support at both sites. Everything all right? Oh, someone's joining with their audio. That's okay. Happens all the time. Good now? Awesome. And we'll try and keep to breaks, but today is really about discussion. So all ideas are welcome. Put it in the chat if you're more comfortable. Raise your hand if you're more comfortable. We'll try and monitor that. I'm terrible about monitoring that, so hopefully my friends at different sites will say, hey, Noel, there's someone raising their hand if I miss it. But we have a lot to cover today, so let's dive right in. Remember, our objectives. We want to present the landscape, which we mostly did yesterday, but today we really want to establish community standards for reporting these PEG lists and actually really define what the hell they are. Excuse my French. How they are and how we're going to use them. Ensure that PEG lists with their support evidence and relative metadata are fair and accessible to the community, and define strategies for evaluation and collation. We want to identify any challenges, right? Like, what does it mean to actually contribute something to a resource for a contributor, and what are the challenges in updating those lists? I think we identified some of those yesterday, but I think that's really important as we figure out what are the standards. Are the standards something that someone can iterate over time, or that someone can easily apply to their list so that they can make it easy to contribute? Then we want to incentivize this. We want to make this easy. We want to think about how to navigate things like journals, our communities and our funders to say, hey, you really should use these standards. These are important. These are helpful. They make it easier for you. That's really the goal. So our outcome, so the reason this all matters, like let's think like a little bit outside of us as contributors and builders of these lists. This all matters because people actually use these. And as far back as 2022, which seems a while ago, we have colleagues who actually used some lists that existed at the time. They're available in the early versions of the PEG KP on the Knowledge Portal for diabetes. And this was to identify a set of genes that they were going to take into CRISPR screens, right? So it's an incredibly expensive experiment. It's like they're kind of banking all their effort on like, what are the best genes to pick to study for beta cell function and diabetes risk. So they actually did this in the paper. They took the 3 lists that were publicly available, they merged them, they kind of did some munging of their own, and they came up with 20 genes based on the heuristic model, the one that was heuristically based, one that was a bioinformatic predictor, and one that was kind of a combination of both. And independently inside that, there was what we were talking about yesterday, where there was like one of the same lists use the other same list thing. So there's a little bit of circularity. But in the ultimate end, this actually produced a biological mechanism from one gene. So this gene was like considered possible, but because of the evidence sources, they chose it, they bumped it up in their list, they put it in their screen, and they made a mechanism proposed for how this contributes to beta cell function in diabetes. We have to do this for lots of genes and lots of phenotypes. So we need to make this easier. This is a lot of work for somebody who actually is an experimentalist who wants to spend their time doing really good biology. We want to make it easy for them to use. So that's one sector. Now, just thinking about that as we go on forward, like that's the type of person that we really want to target. We also want to target someone like Eric Foman, who's going to consume these for, target identification for his company and so on and so forth. So our outputs, you as stewards, you are now more educated. You're going to help us spread the word and make this accessible to the broader community. We're going to try and we're going to make a white paper for sure, but hopefully that will lead to a manuscript that will help provide standards and help journals sort of say, hey, this might be really something you want to adhere to help you make your list accessible. And ideally an ultimate resource, both for the deposition and access to these lists and to presentation and curation and kind of interpretation. So how you can make them useful. And finally, I think today over the course, what's the roadmap beyond doing this? If we are successful in developing these standards and successful in incentivizing the community and successful in building a resource, I think all of those can happen. What's next? How can we make this more useful for the broader research community in the sectors of AI, in the sectors of experimental biology, and so on and so forth? So to kick us off, Laura's going to take the comm and talk about GWAS Catalog's experience. Remember, they did this and they set the standards for how you submit GWAS statistics to a resource and make them accessible. I think we should make sure that anybody who does a GWAS has an effector list. They do essentially. If you're doing a GWAS, you're essentially trying to find candidates. So why not make that part of the deal? So that's one thing that we could actually really think about. Just very simply say, like, if you did a GWAS, you got to have an effector list. And here's how you do it. And then Maria is going to talk about how we presented this so far. So we have about 170 of these that we've incorporated. We have access to them on the portal, fully baked, so to speak. How do we make those useful? How have you done that? What's your experience there? And then we're hearing from Open Tarkins about how they've done the same thing in that respect for their gold standard lists of genes on their resource as well. And then we'll flip back to open discussion. And this is where we talk about the survey that we've sent. And so we pull that back to what are the types of questions that we want to discuss and how do we actually structure this list? What are the key features? What should be displayed? Who do we target? What matters to the folks ingesting the list? What matters to the folks using the lists? Then we're going to pivot to Jason. And I'm really excited about this because this is about thinking about the roadmap. So applications beyond generating these lists, how are they useful to others? We work in sectors like the Common Fund, where basically one of the major outputs of many of the resources is gene sets, lists of genes that do things like molecular signatures. Wouldn't it be amazing if all of the efforts that we pull from these resources for effector lists can be applied to things that are coming from all of these large-scale resources like GTEx, HubMap, MotorPack, all of these, the V2F consortium, where they're pulling all these things together to say, like, these are the gene sets that we believe are active in molecular function X, Y, Z. How could you map them with the effector list we pull from all these phenotypes from human genetics? So Jason's going to speculate and think about some of the opportunities there. So that'll be really fun. And then comes the straw man. Time to actually sit down. Laura and UA are going to walk us through it. Here's a proposal. Here's a place to start. Tear it apart, build it back up, tell us what you like, what you don't like, but hopefully to give us something to adhere to so we can build off of what we think are the standards. And that will really form the basis of our breakout sessions and discussion. So with that, our job will be to go to the breakout sessions, discuss those things, read out together, wrap up, and then really make sure that we follow through on the work that we've done in these last 2 days. So that was a lot to ingest, but hopefully that gives you like the level set for today's conversation. And I'm going to turn it over to Laura, right? Any questions before we dive in or comments? All right, superb.
Okay, can you hear me okay?
Yes, we can.
Yes.
Excellent, okay. Then I will begin. So yeah, I'm going to talk today about our experience at the GWAS Catalog of developing standards for GWAS summary statistics. I'm going to try and whiz through this quite quickly because there's some stuff I want to talk about at the end that I think is really informative for this meeting. So the problem we're talking about going back to 2019, the problem was that not enough authors shared GWAS summary statistics. They would write in their paper that you could access the data if you got in touch with them, blah, blah, blah. And of the thousands of GWAS summary statistics that were shared, they were often lacking data content. So they might just be a list of RSIDs and p-values, which wasn't sufficient really to do the kind of downstream analyses that people wanted to do, like generating polygenic scores and doing Mendelian randomization. There was a lack of sufficient metadata, and the metadata wasn't standardized, and so interoperability was a huge issue. And this was somebody else's paper where they analyzed 327 summary statistic files and found that there were 127 unique formats in them, which makes it very complicated for downstream analysis. So the process that we went through was we convened a group of people, around 50 people, in a meeting kind of similar to this one. So we identified the different stakeholders in the generation and use of GWAS summary statistics. So tools developers, methods developers, data generators, data consumers, also representatives from cohorts and journals and funders, and we set about discussing the issues. So the workshop topics were data content, incentivization of sharing, data update cycle, infrastructure needed to facilitate this, FAIR, which I'm going to talk about a bit later, findable, accessible, interoperable, and reusable, and a forward look to think about whatever standards we came up with were appropriate to accommodate changes in the field in the future. So we had some great discussion about all of that, but really the data content was the one that we needed to nail down to identify a standard format that we could implement in the GWAS Catalog and other resources. So that needed a bit more discussion. So we convened two different working groups. So a smaller group of people, there were around 20 people in each. So firstly, to talk about the file format and content, to identify the mandatory fields, the actual format that the files should be in, any other optional content that we would like to standardize, and issues like variant identification. But we had a second working group to think about kind of higher level issues around privacy and diversity. So we wanted to make sure, it's a great thing to set standards and insist that everyone follows them, but we didn't want to make it impossible for people from under-resourced settings then to share their data. We didn't want to put barriers in place. And we also wanted to consider that, some people are working on sensitive phenotypes and might have legitimate concerns around privacy and de-identification risk. So we really wanted to make the set of available GWAS summary statistics as diverse as possible. So the first working group met 3 times, and we also did an online survey, which was kind of asking the wider community what they thought about the proposal for mandatory fields. And each time we iterated the standard and reviewed it and refined it and identified areas where we needed to discuss. And similarly with the diversity group, we had 2 meetings there, and in between we iterated the plans. And we also went out and talked to other stakeholders, for example, the cohorts who might have issues around controlled access requirements and so on. So the outcome of all of that was that we published a paper in bioarchives describing the standard. We also published the standard on GitHub, and we asked people, so that wasn't the end product, that was the outcome of the workshop process, but that wasn't the final product. So we asked for more feedback on that, and we put it out to the community again, and we did some more iteration, and we got some really good feedback at that point. From big things like we hadn't considered sex in the metadata, and somebody pointed out that there were about to be released a lot of UK biobank summary statistics that were sex stratified. So it was really important to be able to describe them properly. And also small things like using NA for missing values. That can also be a valid nucleotide string. So we changed that to hashtag NA, which was very easy to do. So following that, we'd finally arrived at the standard and we wanted to implement it in the GWAS Catalog, in all of our infrastructure, which I won't go into details about, in our harmonization pipeline. We also developed tooling for the standard. So we have our own internal tools for formatting and validation for users to prepare their files. We collaborated with Plink. So Plink now has an option to produce files in the GWAS Catalog format, ready for submission, and also to produce documentation and training materials and so on to support users to share their data. So the outcome has been really significant. So there are now more than 85,000 summary statistics files available in the GWAS Catalog. We had a 33% increase in submission rate after we published the standard. All of the new submissions, except ones that have non-standard study designs, have all the mandatory content. So things like effector allele frequency, effect size, and things that were missing in around, well, at least 20% of files prior to implementing the standard were missing those important data fields. And we also defined optional content. So we didn't want to make it impossible for users to produce data. So we have minimum requirements, but we also have encouraged fields in the data and the metadata. 75% of the data files contain additional content that is non-mandatory, and 85% of the metadata submissions contain additional content. So we felt like we'd struck the right balance there between mandatory and optional content. And something I didn't actually mention on the file on the presentation, but there was a paper published last year by a group at Cambridge University showing that the citation rate for papers that shared data in the GWAS Catalog was higher than those that didn't. So those are all really great outcomes. So I just wanted to finish up by talking about FAIR, so findable, accessible, interoperable, and reusable. And we think about this a lot as users, and this really informed the approach we took to defining the standard format for summary statistics. I think we can really apply this to the discussions we're having today around effector gene lists. So thinking about the different indicators, so to make GWAS data findable, in the GWAS Catalog in general, we have a unique identifier for each study, by which we mean each data set. So not just a publication, but each phenotype that's been analysed within a publication has its own identifier. We have rich metadata for all the studies, and all the data is searchable. So it's really easy to find that data. And it's also accessible, so it's open and freely available via a web interface, downloadable spreadsheet, and computationally via our API. So thinking about the summary statistics files, we decided that in the workshop that each summary statistics file should be linked to an GCST, which is the accession ID we use in the GWAS Catalog. And those should also be searchable by the study metadata. So you should be able to search summary statistics, for example, by ancestry as well as by trait, potentially whether they're case control studies, all of those kind of things. They should be linked from the main GWAS Catalog pages, so you can really easily come into the GWAS Catalog and search for a trait, find summary statistics linked to it. And we also have another page that lists all of them. So if you want to browse, you can do that as well. Summary statistics are accessible. So again, they're freely accessible in multiple ways. And an important point as well is that we store the study metadata separately from the summary statistics files themselves. So if summary statistics need to be removed, we can maintain the study metadata. So that's always trackable. So thinking about peg lists, so I had to think about how we could make peg lists adhere to the FAIR indicators. So again, I think we need to have a unique identifier for each peg list. And what we consider a single list, I think maybe we need to discuss that. but they definitely need to be accessioned. And again, they need to be searchable by study metadata, and that means the metadata needs to be standardized and structured. They should be linked from a central data repository, and as Noël already mentioned, that's the kind of overarching goal here. And they should be linked to the source data in the GWAS catalog. And again, this is kind of connected to what Noël just mentioned. So if you have a peg list and you can provide the accession ID of the GWAS, we can really easily link the two data sets together, and that makes them findable in either direction. And they should be accessible in terms of being open and freely available, and also with persistent metadata. Thinking about interoperability and reusability. So in the GWAS catalog, we make the data interoperable by using commonly used controlled vocabularies. We use the experimental factor ontology. We use standard nomenclatures for variants, genes, and regions, and we have a controlled vocabulary to describe ancestry. So the data model is very well defined, and the data is reusable. We have a clear data usage license and detailed provenance. So every data set is either linked to a publication, or it has the detailed provenance of the submitter. So in terms of summary statistics, we thought about the standard format, the publication of the standard format, and also we provide harmonized files as well as the raw files that make them more interoperable with other data sets. So we've done all that work for our users before they get to the data. And they're reusable, again, they're licensed. We provide detailed provenance and enough sufficient metadata to make sure that the data can be reused. So thinking about the peg lists, again, what we need here is a really well-defined data model. And obviously that's something that we're talking about today. We need to be using controlled vocabularies, especially for traits and evidence types, and standard nomenclature that was already mentioned for variants, genes, and regions. And then again, if we're thinking about representing them in a resource, there should be a clear data usage license, detailed provenance, including links to the original GWAS, and we need to define what the mandatory metadata is to allow this kind of data to be reused. So that is it from me. I'm happy to take any questions or follow up at any points.
Virtual and in-room clapping for Laura. That was awesome. I think, go ahead, Maria.
I have a question. Just curious, like from the time that you realized that the standardization was needed till the time that you feel that this is all mature and users are using the system, how long did that take roughly?
Well, so I think we started talking about this we started accepting summary statistics in 2018, and I think that was probably the point where we really thought, you know, we need to sort this out. And open targets has been a big part of that. I should mention that as well. Sort of pushing us forward to do this, because the summary statistics are a really important output to the open targets platform. So yeah, it was sort of 2018, 2019, we were talking about having the workshop. The workshop was held in 2020, and then we finally implemented the standard in 2023. So that's kind of 5 years from talking to actually having it working in practice and accepting submissions. I think we probably could have done it quicker if we had more, you know, more resources, more funding, all the rest of it. But yeah, we got there.
Yeah.
And without a pandemic in the middle as well, as Eve reported.
That's right. Yeah.
Details. So I have a question. I've thought, so I'm looking at this, and you know, as a person who works with folks that submit GWAS data, GWAS is kind of almost simple, right? In the sense that, like, maybe that's because we're over the hump of how hard it was to get standards. But you know, there's not a lot of files, and it's just like there's very specific data types that you're sharing in the metadata, right? There's like metadata, there's like allele frequencies, p-value, there's the summary statistics, blah, blah, blah. With these effector lists, we started to see yesterday, you know, from Eric and from Adam and from Cassie and from everyone that was presenting, there's lots of human curated content. There's a lot more lines of evidence. So it's more building like a case against a gene, so to speak, not against, but for a gene. So a totally different sort of type of file. And it's not really a list, as I think Jakob pointed out, it's kind of a matrix. And it's a bit of a combination of very clear, like some outputs, like statistical outputs, but also like evidence sources, like data was found here. And then almost like a column of like, this is what I believe and this is why. I mean, there's some human readable text. So I think as we think about, I think you did a great job actually, Laura, laying out how it was done for GWAS Catalog for summary statistics so that we can easily sort of like lift over the things that are simple liftovers, right, into peg lists. But how do we then think about the exposure of the other things that need to go into those and the ability to make those more useful and standardized? I think that's complicated because I guess I would ask, here, I guess the questions, like folks like Cassie and Adam, you've done both. You've submitted data to GWAS Catalog and you've also submitted peg lists to the knowledge portal. And I would say we worked with you, Adam, for quite some time. If Adam's on, we worked with Adam for quite some time, sort of figuring out what do we ingest? What do we represent? How do we curate it? How do we make it accessible? And that was a process. Whereas his GWAS catalogs, they just go in. It's almost like paper, go in. So we're way ahead with that. But is that because the complexity is different or because you guys are just way ahead with the standard, do you think?
I was talking to people yesterday about the process and how we kind of iterated it and how we almost came to a point where we tried to make our, one of our supplementary tables directly ingestible into the portal, right? So part of that process was almost systematizing or almost coming up with, I guess I say it now a bit like when we try and submit to the GWAS catalog our summary statistics, you have your Excel metadata spreadsheet that you need to kind of fill in for all this stuff. So it is more complex, but I think actually some of this process, could still be there, right? And that it's just working out what the fields might need to be. It might need to be more flexible, I guess.
Yeah.
Because although there are 127 different formats of GWAS summary statistics, you could probably boil that down to a relatively small number of relatively standardized variables, I guess. Whereas here, people can use anything they like to make a peg list, right?
Right.
I guess my question thinking about this is, now I guess as a field, we move towards making summary statistics available, partly because for some people it's the right thing to do, partly because journals told us to, etc. The carrot and stick. And I guess then you bring that next layer in of you develop the guidelines, and now I want to put my summary statistics in GWAS catalog. Oh, now I need to go through this pain of filling in this spreadsheet to do it. I'm doing this because I want the summary statistics to be available, but I have to, there's a kind of gatekeeping process to it, which if I'm honest as a submitter, particularly when I'm doing it for a GWAS of 250 traits, I'm probably going, oh, I wish I didn't have to do this, but I know it's going to be beneficial for the community, and GWAS Catalog wants me to do it to put my data there. So I'm gonna jump through that hoop because I need to, right?
Yep.
So I think one question here is about the sort of gatekeeping of this. How do we? Firstly, I don't think we yet have the community drive of you must make peg lists available, partly because we have the portal, but we maybe don't see, oh, I'm writing a GWAS paper, you must put this in somewhere. So I think there's got to be that bit, but then also we need that gatekeeping bit. That's what's going to force people to, especially because it's going to be harder, as we're acknowledging. So how we think, and that might be that there's multiple places where you can put peg lists in the way there are with GWAS summary statistics, but kind of, I guess as a community, we're going to have to encourage people to submit them, but we're also going to have kind of have that gatekeeping, I think, to make people start using it, to force people to do it, to fill it in, to get the recognition or to get the kind of content there that will help it snowball, I guess. So.
That's exactly what I wanted to hear from you. I think that teaches the fact that I guess GWAS catalog still is hard. There's still some hoops to jump, which actually is in a way good to know because it means that like you're willing to go the extra mile, right? So I think there's a hand in the room, but I think, so go ahead, I'll stop because I want to hear what other people have to say.
I think that's my hand. Thank you, Ellie from Open Target. I was also thinking, I think with the FAIR elements that Laura outlined, there's elements there under accessibility and interoperability that I think apply very much to the peg lists, and we can, like we're saying, we can sort of easily extract those kind of rules and guidelines across. But I think things like the fair aspects of having, say, an accession ID for a particular peg list may be tricky, because it's not necessarily going to be static. If we go for a framework like the Gene Curation Coalition, where, you know, people are potentially submitting their own views, their own opinions of which genes they think, or their own, you know, they've got their own contributions from methodologies they've used to analyze. Those lists may, you know, be constantly being kind of re-evaluated. The evidence for those is kind of changing, and those lists may change. So even if you ask, say, submitters of a GWAS study to submit their effector gene list, as that GWAS is then reanalyzed potentially with, you know, new QTL data sets, say through our Open Targets L2G pipeline, we may come up with a slightly different effector gene list. But I think it's that might be something tricky of having, you know, how do we factor in the having a flexible or ability for these gene lists to change with growing evidence over time, and necessarily be static and you know.
Yeah, and I think that's going to be a really important point for the discussion later.
Cassie, you have a question or comment?
Not so much a question, I was just gonna sort of extend on what Adam said. So, you know, as someone who submitted to the GWAS catalog in the older days and more recently, you know, the hoops you have to jump through now, or sort of that gatekeeping exists, and it is harder to submit your summary stats, but because the journals require you to post GWAS summary stats somewhere, you sort of don't have a choice but to go through these hoops if you don't want to, right?
Right. So
I worry with a peg list, if this is not something people are like have to share, we're doing it out of the goodness of our heart or because we want it to be out there. If we make it have too many things for them to do, that they're not going to do it.
Yep.
And so I think that's something we really have to think through when we're putting this together, because I don't see it being something that people are required to do, at least not anytime soon.
Yeah.
Agreed.
I think those considerations are part of like how, so it's almost like the reason that we came together with the GWAS catalog, we as a knowledge portal, we think about the fact that I'm going to put Maria in the position, having a curator who's interested and cares about that, or like staff to think about this, that partner with the submitter. Now, obviously, this is not necessarily scalable, but it's worked in some regard because it's taught us like what are the things that you must have, right? They're bare bones in some respects. And then some of the things that you need a human to kind of work with that submitter to say like what matters. So this gets to Ellie's point of the idea that it is iterative. So it's an interesting, great, like maybe there's like almost a continuum we can consider, which is there's some bare bones that make it so that when someone's submitting a GWAS, they submit the basics of the list. And then there's more for the presentation, the curation, there's like extended stuff. We can consider sort of like 2 tiers as we go to submission and reporting to allow people to have the option. But I mean, frankly, if you go to the extent of doing a GWAS, your goal is to find genes that cause disease. That's like why you're doing this in the first place. So if we do this really well, this kind of can fit with the process, and it will be a little extra pain for people, but hopefully it will make it more useful to the broader community.
Yeah, I think we need to move on, but I wanted to make one final point that about the metadata. I think is the real thing where having the standards in the GWAS catalog has made a difference, because you would find, the file formats themselves would be, you know, there's a relatively limited number of variables that are included there, even if they've got different nomenclature. But the metadata would have just been written in a paragraph in the paper. And I think that's where very analogous to the peg lists at the moment, you know, to describe all the different experimental data that's gone into creating a peg list, that will just be written out in the method section of a paper. So to have that structured in a way to make it searchable and findable, I think that would be a huge step forward.
It's a good point. Actually, it's a great segue to presentation of these lists and what it takes. And so, Maria, let's turn it over to you, shall we?
Yes. Okay, so there we go. All right, so as I mentioned yesterday, starting in 2019, when we first implemented the Mahjong McCarthy peg, peg list in the knowledge portals, we started collecting other peg lists, started looking for them and collecting them. So I just want to quickly go over how we do that now. I think this is all really going to change after the results of this workshop. But at the moment, we've just been trying to amass them and get an idea of what they're like. So we've just been finding them through, of course, the GWAS literature. That's where you find them, and we look at GWAS literature to get genetic association summary statistics anyway. So we see them there. We also run some automated PubMed searches that are mostly geared towards finding GWAS again. And then often authors come to us and say, "I've got this list, would you implement it?" And so in some cases, we've worked with authors to get the data. In some cases, for example, where the list is graphical only, some authors have supplied data in table form. Sometimes we need to talk to the authors to actually understand what they've published, and we've done that. And then also we get their advice on how to represent the evidence in a logical way, and I'll show you what that means in a couple minutes. So we end up with a comma separated values table of data, and we load that to our bring your own results platform. So there's a CSV file at the top, and then the table is configured by a JSON object. And I just wanted to mention this platform because if you're not aware of it, this is something that anyone can use to upload your own data and represent it in tables and visualizations. So if you have data you want to represent, take a look at it or get in touch with us. It's a nice thing to use. So when we represent these results in the portals, we have a free text representation of their provenance. We know, of course, with the reference and the evidence types that are used. And then for some of the lists, we have an additional section called the research method, just with more details. And I started out doing this for every list, but it really became too much work. I just couldn't make any progress by doing it representing every list this thoroughly. So the ones that have research methods now are usually the ones where we've worked with the authors and they've supplied all this detail. So with this BR system, we can create nested tables of the data. So the top row of the table that you see by default just shows the most important summary information. So here's an example where we've got the locus, the predicted effector gene, and in this case, it was given a priority score. So that's kind of at a glance the information. This table, you can also add filters to filter and set thresholds for any of the values, and you can also sort the columns by just clicking on the column header. So these rows, when you click the view button, they're expandable, and all the details are here. And this is showing the details for a really rich, data-rich peg list. But so, for example, we talk to authors sometimes to get their advice on how to organize all these types of data. So like these are kidney annotations, and then there's QTL values and so forth. So by expanding it, you can see all of this. Yeah. We make all the lists available in our predicted effector genes knowledge portal, or PegKP, pegkp.org. And on PegKP, we have a list of the lists. So this is sortable and filterable. You can search by author name or by trait and so forth. We were trying to get peg lists for most traits or most trait groups, and not surprisingly, most of the ones we have are for the cardiometabolic traits that we usually curate, but we're trying to get them for as wide a range of traits as possible. We've also implemented a search by gene feature. This is in PegKP. You know, you might have a gene of interest. You want to know whether it's on a list or not. But as this issue has been brought up before, some genes are on many lists. So this is GCKR, which is, as far as I can tell, it's a gene that has got its finger in every pie, and it's on 50 peg lists. So that's not super informative. So I think, and of course, our peg lists are a mixture of the lists that have the top gene only and the lists that have all genes. So GCKR, some of these lists that it's on could be ones where it's not the prioritized gene in locus, but it's in the list. So that this is clearly not the optimal way to present this information. We also make the PEG lists available via the gene page of the knowledge portals. And again, here's an issue with the different types of lists. So initially we displayed on the gene page every PEG list that the gene was on, but that was for GCKR, that was a list of 50 PEG lists. That's not, that takes up too much real estate on our gene pages. So we changed that to just show the lists where this gene was the top gene. Which is good, but you need to know. We haven't sorted this problem out yet, but this is not providing full information. I think more standardization is going to help us direct people to the PEG lists of interest much better. We also have PEG lists available through our Gene Finder tool, which is a tool where you start out with a trait of interest, and then you can add all kinds of information to this table, including expression levels in different tissues, common and rare variant associations, and then presence on a PEG list is another criteria that you can add to this table. So, yeah, I think, in general, we have just tried to upload as many of these lists as we can, and we've just taken them in the format that we find them. So they're completely diverse in format, I think it will really help to get more standardization, and we'll probably want to rework the list that we have, or at least change our system going forward. And that's it. And for this, I just want to acknowledge our wonderful team who implemented everything that I just showed, and our colleagues at EBI and UCSD and the University of Michigan. It's really a great group. Thank you.
Thank you, Maria. Questions for Maria, or comments, or has anyone actually used the portal to play with these lists and has reflections? I know I have, and I've been frustrated every time. Because I do think that it's really tricky when you have like a gene that appears in many lists. That could say something about biology, or it could just be the fact that the way that we've curated these, like as Maria said, that they annotated every gene in the genome with a list, and obviously appears in that list. So there needs to be some way to also have that we as a resource would do, if we had all these lists done standard, to present them in a way that helps you make sense of them. Like I've always had this vision where it's like, could you then take all the lists that are in the portal now and match them with some of the huge calculation scores and trying to develop, I don't know, pathways that fall out of it for given phenotypes, or intersect with phenotypic correlation. And there's a lot of things you could do. So I think as like a person who wants to use these, or as a scientist that wants to help others use them, that's what I'm thinking about. So, we're a long way from that, but, I think we have to be keeping that in mind as we go forward.
And I just realized I completely slipped my mind, but we also represent these lists on the phenotype page. So that's another, I think, a good way in general for people to access them. You're going to want to know about a trait, whether their PEG list is available. So
right.
we do have that.
Yeah, I can ask a question. So do you find you can curate all the data that's included in all the PEG lists, or is there some that has to get dropped out because it doesn't fit into the data model that you're using?
I'd say we can curate. I mean, it's really, if it's represented in a table in the paper, we can re-represent that table in the portal. So I think that, yeah, it usually works.
So are the limits of what you can curate kind of like you were saying yesterday about if there's just a figure with some dots on it, you can't ingest anything from that? Whereas if there's some data that's nicely presented in a table, you can curate it.
Yeah, and I would say that the major difficulty, what takes a lot of time, is actually understanding what has been done, because it's often really not explained. So just chasing down what, there may be a header in a column in a supplementary table that doesn't mean anything to me. It's not, and you might search the paper for that phrase and it doesn't come up. And so, what is this? It's just stuff like that.
Yeah, I think making the analogy to the GWAS summary statistics as well, I think this is something where if it's the data generator is putting the data into a format themselves, it can actually be much more productive, because like we know as GWAS curators, sometimes we're hunting around in the paper for the simplest of things that are kind of obfuscated by the language and the number of edits that have been done when they were writing the paper. Whereas for a submitter, they know what cohort they use, they know what the ancestry is, you know, they know whether it's a case control study, they know how the case control numbers match up with the ancestry, which is one of the big headaches. So obviously I don't know this for sure, but I assume it will take the data generator a lot less time to fill in the metadata form than it does for the GWAS curators to hunt out the data from a paper.
Exactly.
And that with the peg lists, hopefully it should be the same.
Agreed. All right, in the interest of time, should we go on to Jack from Open Targets is next, I think, right?
Yep.
Awesome.
Hello, can you hear me? See my screen?
Yes.
Yeah. Okay, great. So yeah, so we're here from Open Targets to kind of see how we can contribute to this predicted effector gene knowledge portal resource. Obviously our work, we don't directly curate these gene lists ourselves. We have a kind of model that predicts these for us, but obviously we can walk through how we curate the effector gene lists that we do use, what kind of additional annotations we add, and explore some of the challenges and where we can possibly contribute. So if I break down the title of the talk, you know, curation and display of the gold standards, there's really two sections to this short 10-minute talk. First, the curation part. So where do we get these curated gold standard lists? And then as far as curation goes for us, that really means what additional information do we add to these existing gold standards? And then moving on, we'll kind of talk about display. Yakov already touched on this yesterday in his talk. So how do we visualize the final output of the predicted genes from our model? And actually in terms of the predicted, in terms of the peg list itself, it's very similar to what goes into our prediction model in the sense that it's very similar to the feature matrix that we use for machine learning. And then we'll just wrap up with the summary and the discussion points of everything. So let's jump in because it's 10 minutes. So this is the kind of input gold standard gene list from the original publication for the L2G model back in 2021. So you can see it's made up of a variety of sources, expertly curated, or some with functional validation, some not so much. And we also added in data from Campbell, so that's drug data, drug targets that have reached high clinical phases that we can use as a proxy for potential predictor effector genes. We haven't actually updated any of the expertly curated set of genes, but we have made recent efforts to expand our gold standard list from Campbell. So because there's been additional Campbell data releases. And these are the list of gold standard positives. I will talk about the gold standard negatives later on because there was some interest in that yesterday in the talk from Yakov. But for now, just imagine at any given GWAS region, if we have a gold standard positive, then all the other genes are gold standard negatives. So I'll walk through the basic schema of what a gold standard or predicted gene list looks like for us. The bare minimum we essentially need is for you to tell us the disease or trait that's related to a gene. So that means, give us the disease or trait name, the EFO ID, the gene ID for that. And then, of course, everything else would be nice to have, but not absolutely necessary for our purposes. So I'll talk about how we can essentially just take these few columns and annotate data as we require for those. So just to refresh, a refresher from yesterday, where we showed off kind of the amount of data that we produce at Open Target Genetics. So this is all the kind of co-localization fine mapping results from the previous release we did back in June. So we're mostly focused on kind of post-GWAS analysis. And as you can see, there's a lot of kind of credible sets from EQTLs, credible sets from GWAS. And I'll talk about how we can essentially annotate any given gold standards that we get from these results. So all those results essentially boils down to these three data sets. So the study index is where we keep an index of all the studies that went into our analysis. That's the study IDs, the trait IDs, all the metadata and things that's kind of directly ingested from GWAS Catalog and other resources. Then for each one of those studies, we have a credible set result table that contains detailed summary association statistics for the top hits, the fine map credible sets, depending on whether we had summary stats or not. Then from the credible sets, we perform co-localization across all overlapping credible sets. So just from these three tables, you can see there's a few columns kind of in common, and we can use those to join these tables together to annotate both gold standards and other credible sets that we don't have effector lists on, peg lists on. So the central core element of our product is the credible set, and now I'll kind of talk about the exact features that we annotate those with. So we annotate every single credible set in our resource with a list of different features. So that ranges from in silico kind of pathogenicity prediction to how far away they are to genes, some epigenetic data sets, we include that, as well as QTL co-localization from the vast range of QTL resources that we ingest as well. This is the full set of features that we intend to have in our model, and the top section here are what we call variant level features. So you can imagine for any given credible set, you'll have a set of variants, and each variant will have a different kind of contribution score to how it's affecting each gene. So in this small illustration here, there's a small credible set of two SNPs, essentially, 1 and 2, with different posterior probabilities of causality. And what we mean when we say we weigh variant level features by their posterior probabilities is just that we simply care about variants which are more likely to be causal in a credible set. So we care more about how variant 1 has a VEP score for gene 1 than gene 2, and so on. And then the last section, the QTL co-localization, are formal co-localization tests that we perform, either through CoLoc, we have summary statistics, or through an approximate approach using eCaveat. And although we ingest a lot of different QTL resources, actually in terms of feeding into the prediction model, we only use the strongest co-localization result. So this is definitely a challenge that we face when it comes to visualizing all the rest of the co-localizations available. So to summarize, in terms of our peg list or feature matrix, we compute this for every single credible set in our resource, and we do that for every single gene within the kind of credible set range itself. And briefly, we also perform a merging step in our training step. So what that means is for every credible set to gene annotation that we have, we merge that feature across other credible sets in different studies with the same EFO. And we do that to mainly kind of address feature missingness as well as class imbalance, because just by the nature of being a gold standard positive, you often have more features annotated than more features missing. We've also had quite consistent feedback regarding the kind of class imbalance. So how many gold standard positives to gold standard negatives we have. Usually is at the moment is about 1 to 7, so that's quite high. And we are using the feature matrix to downsample gold standard negatives to balance that out a bit better. And that's done based on keeping gold standard negatives, which have the most features, so minimizing on feature missingness when we feed it in. So I won't be talking about the model performance and things, but I think all might be interested about which features are most important in this model, and its distance. And there's two main reasons. One is that the gold standards are mostly based on mostly include the closest gene. And also the second reason is the feature missingness. So you always have a distance feature for every credible set to gene mapping, whereas you don't have that for the other features that we have. So that's how we present the feature matrix. And then in terms of the result itself, this is kind of a screenshot at a portal. So for each credible set in each study, we kind of present detailed association statistics directly from the GWAS catalog, as well as the kind of predicted L to G score between 0 and 1 for you to interpret yourself. We also have a partial score based on the leave one in analysis for the L to G. So instead of running the prediction model using all our features, we only use a class of features to see what scores come out from that. We're not sure if we want to keep that for the latest release yet, but something we're working on. And in terms of co-localization, although I mentioned we only use the strongest one, there is a detailed view where you can see every single co-localization with this credible set, and it's visualized in using these bubbles, where the size of the bubble indicates how significant the co-localization was, and you can sure, and you can see there's also two different colors. So blue means it's based on the beta direction, essentially. Okay, so out of time, but the summary is that, you know, we're obviously using these peg lists for our machine learning approach. So having a unified database of these will really allow us to incorporate a larger, more diverse set of gold standards that will hopefully let us see an improvement in our model. And we're already very well kind of positioned to contribute fine mapping, co-localization annotations, because we're already systematically doing that anyways. In terms of the kind of L to G score, I did see in the template for the breakout sessions that there were columns related to that. So that would be good to kind of have a further discussion on. Do we plan to use L to G's outputs as an additional annotation, or do we plan to have a peg list that's based on in silico predictions from methods like L to G or any of the other methods that we saw yesterday? For us, we kind of bypass this problem of assigning disease to gene mappings from resources like Campbell, because you don't know which GWAS signal or study to assign that mapping to. So for us, we just pull together all studies that contain that trait. But from the kind of main figure, it looks like we want to have the gene linked back to a GWAS signal somehow. So that is something that we'll need to discuss. How do we do that? Do we do it on a study basis or on a region to gene basis? Things like that. And of course, there's so many QTL resources. The kind of co-localization results in the template, we'll need to figure out how to efficiently display results like that, because we don't want to have 100 different columns for every tissue that has a co-localization, things like that. Yeah, so that's that. I'll just flash the acknowledgement slide and then we'll move back to the summary. And of course, welcome any feedback and discussion on both peg lists as well as kind of our approach for these features. So yeah, thank you.
Brilliant, thank you. We are running a bit about in time, but that's okay because discussion is actually good along the way because people are thinking about things. So questions for Jack, comments, considerations.
All right, let me ask a very quick question.
Yeah.
Thank you. Thank you for the great presentation. Is there a way for us to run L2G prioritization on our own GWAS SumStats?
In theory, yes. So we have released the Gentropy package that will allow you to do that. But the problem is kind of generating the features. So if you imagine the step where you have to figure out which QTLs co-localize with your GWAS output that you have yourself, it would involve downloading all of the QTLs, fine mapping credible sets from our side and running that overlap and running that co-localization step yourself, which is possible. But yeah, so in theory it's possible. In practice, I'm not sure if we've actually tried it recently. Yeah, in theory, yes, it's possible. Yeah.
Thank you.
Thank you.
All right. So I think given the timing, we are going to flip the agenda a little bit so we can save discussion for the end. So we're going to switch to Maria's presentation, I believe. Oh, Satoshi, do you have a question?
Oh, sorry, it's already answered, so thank you.
Oh, beautiful. Okay. Marie's going to summarize what the community, beyond some of the folks that are here, and you all participated and took the time of your day. I appreciate that. But others, had some really great feedback about what they thought we should be doing. So we thought it would be useful to share that now and then pivot to a discussion. And then we have Jason's talk and then we'll go to breakouts. All right, Maria.
Okay, so yeah, about a year ago, we started, spurred by the success of the GWAS Catalog standards development, we decided a survey would be really good in this area as well. So with Aoife and Laura and Yue at the GWAS Catalog, we started working on a survey and designed it, and we put it out in the spring, and we had it open from March through April. So I just wanted to present an overall view of the results. And there were a lot of nice free text comments that I've excerpted in here, and I'm not going to read them all now, but these slides will be available. So you can all take a look and see what people had to say in detail. So we got 58 responses. Many of you on the call responded, and thank you very much. There was a geographical distribution, not huge, but not more than the US anyway, and Britain. And we had a good distribution of different level career stages, from PhD students to professors. We had representatives from NCBI and also from a lot of industry folks from different companies, and that was nice. We started off asking about terminology. What should we even call these genes? And we gave lots of possibilities, but so causal gene and effector gene were the top two choices. But then a lot of people had really good worries about using causal, and people have said that journal editors will catch you on that because it's too strong. And I think generally people see causal as being like functionally, experimentally established to be causal, and that doesn't apply to these predictions. So in our group anyway, we like effector gene, and we're going with that. And we asked people how would they use these lists, and many of them, in fact, the most popular answer was as an input to bioinformatic analysis or a pipeline. So that just highlights the need for standards and interoperability here. Which genes are your, which lists are you interested in? Most people said they would be interested in a list for their disease of interest and the related traits, but then some just wanted any list for any trait. And I think maybe those are the ones who would be going on to develop bioinformatic methods. Where would you go to find these lists at the moment? Most people would just go to the literature. Open Target's got a lot of write-in codes here. These are the free text answers. Somebody thought ChatGPT was a good source. I'm not sure I agree with that, but okay. What are the barriers to using the lists? Well, the transparency and provenance were big ones. How do you know what people have done and where do the data come from? And lack of standards for confidence and ranking. I think we're converging on those issues here too. We asked people to rank how valuable different kinds of lists were, and we gave them options for bioinformatics derived, manual curation derived, combination, and with single or multiple lines of evidence. And the real winner was that they liked a combination of bioinformatic and manual curation and multiple evidence. We asked, should these peg lists based on GWAS loci be distinguished from lists of genes that were generated in some other way, like candidate genes? And most people said yes, they should be distinguished. This question was about scoring, and most people said they would prefer numerical scores for text categories, although quite a few people said that they prefer no scoring categorization. We asked whether the range of genes, so this was a question about whether the list should present the top gene only, the single most likely effector gene, or the evidence for all genes. And this was striking. Most people really said they wanted to see evidence for all genes. And then there was a comment here that I won't read, but it's I think it's very, this isn't the only time that I heard something like this, but someone says that they had all the in silico evidence pointing to a certain gene, but then the experimental evidence showed that just definitively was not the causal gene that represented a different gene was the causal gene. We asked whether people would want to compare multiple lists, and most people would, either for the same trait and then for cross traits. I'm not sure. I guess that this means if you whether a gene is on lists for multiple traits. Would you like to be able to interact with the list and assign different weights to the different evidence categories? People like that idea. They would like to interact. And then these are just some free text comments that are interesting, and I will share these. I'm not going to go through any of them right now, but it was really nice how thoughtful everyone was in answering this survey.
In terms of the comments, what were some of the key takeaways, Maria, that you felt were like really important to make sure that we address?
Well, I think that to me, the most striking thing to come out of the survey was that we really do need to present all the evidence for every gene. That just a single effector prediction could be wrong because we don't have the right type of evidence for that gene or something like that. So I think people recognize the need for that.
Right. And this brings us to the point that I think I'm seeing in the chat and over conversations is that it's not just a list, like here's the genes, thank you. It's a matrix. It's almost more of like a nested table of evidence sources based on different types of things, whether they're bioinformatic predictions that come from lines of evidence, as you said, Maria, sources of data, or actual just human curation and summaries. So that whole matrix of things is something that we must, and also because of these different tissues and so on and so forth, so lots to consider. So I think that's really, it echoes what we've been talking about today from what you said, and it also really does stipulate that building a resource that captures these types of things is critical. All right.
So, question.
Yes.
Maria, what are your thoughts on the word candidate?
Well, to me, that implies the older studies where people would have a list of candidate genes because they thought they were involved in a pathway that was important for a disease. It was to me, that has nothing to do with GWAS. And Julie, do you have comments from the rare disease
perspective. So candidate is fairly commonly used in rare disease. I don't know, I guess in thinking about most of those cases, they are biologically driven, but yeah, so I think maybe having it, I don't know.
Well, I'm thinking about the pushback of causal, right? Causal implies validation,
Yes.
at least for me.
Yeah.
Whereas what we're doing is building a matrix of evidence with some number of columns, and you get a check mark, if it falls positive or negative.
Mhm.
So it still remains a candidate. And if it's a gene-rich region in high LD, there might be a lot there, right? So it's unlikely there'll be multiple causal genes, although not impossible. It's likely the causal gene is hidden among many that still survive the triage. And so I'm just wondering whether the concept of candidacy needs to somehow be incorporated into these peg lists.
Some people use candidate causal as combination.
Yeah, I mean, we're always careful to say predicted effector gene, right? Not effector.
Correct.
It's the effector gene to me implies the causal gene, so we don't want to call them that. Yeah, I don't know. It comes kind of down to semantics at some point, but yeah, that's an interesting thing to think about.
All right. I think we have, let's concur to the last talk before I break, I think. Yeah. We're going to have Jason Flannick speak now. And the reason we asked Jason to talk is, there's a lot of things that he's working in this space that are relevant. We talked about those yesterday with Pigeon and things like that. But what I really want to have him talk about today is thinking about the consumers of these lists, whether it be a chat bot, no, I'm just kidding, but AI and generative models, knowledge graphs, but really about the users at the other end of these lists. What's important? What can we consider there? Whether they're biologists, whether folks in industry, whether they are other resources like open targets as an example, who would use these in other settings to make them useful for another context or in their resource. Because ideally we want these to have life beyond their initial publication or initial sharing and be useful in comparison and contrasting and overlapping and also generating hypotheses across effector lists that come from human genetics across traits that may be related. This is all critical, but we cannot do this without a way of standardizing the representation of them. So Jason's going to talk about some of that and I think really help us as we discuss the straw man later.
All right. You can hear me?
Yep.
All right. All right. So just as a disclaimer, this is actually not my area of research. AI and computation. Well, I work on statistical genetics methods, but in terms of standards for how these lists might be fed into them, I would not claim to be the lead all expert here. So I think you can view this mainly as just some topics for discussion later. And just some, I was just thinking through any changes or any additional requirements if we would like to standardize these lists. If we think about a user who's not, I think the implicit user we've been talking a lot about here, which are, individuals who are going to like view these through a portal or like almost knowledge. And instead think about the larger scale consumption of these into downstream methods. So I think it was actually, I hadn't seen that survey result, Maria, but it was interesting that the majority of people, it looked like, would want to use these lists in these downstream methods. And so I think it's important to think through that there could be some different requirements in serving those users as opposed to like biologists, things like that. So I was just thinking through, just to set the stage, what actually do we mean when we say a computational method that would consume or would use these lists? So here, we're not talking about generating a method to generate these lists. We're talking about once a list is produced, a method that would consume them. So one thing I would say is that they're not really big data, in the sense big data being like large enough to do like classic data mining or inference on that, right? Because the most you're talking about would be on the order of 100 or so genes for a given trait. You might have 1,000 if like with predictions, but maybe if you start looking across many traits, you might have that, but then you're getting into heterogeneity of the input. So I think it's more reasonable to consider the use cases of these in a more narrow sense. And these are 4 that I came up with. So people might use these as truth data for training models. Brent talked about that a little bit yesterday. A second would be very closely related would be a benchmark data set for testing models. So obviously the requirements would be similar if you're using these as a benchmark or for testing versus data for training. I think another, moving beyond just the predictions themselves, because there are so many features that are being used, and I think I didn't get the name of the person, but from Open Targets, about building the gene features is in and of themselves like a really difficult and cumbersome task sometimes. So the actual inputs that were used to generate the lists in the first place, I think, could be an important data source that people might be interested in accessing. And that could be because they want to, use this in their own models, or they want to validate or check the model or check sensitivity for the initial predictions, things like that. And then I think in terms of downstream, more cutting edge, like ML, like I was thinking a lot about the extent to which these could be used in LMMs. And I can't really see how these, this curation would do for LMMs better than just what the LMMs are doing out of the box now by basically sucking down the whole internet. And there are others who might be able to talk about that. I know there was that paper that came out of 23andMe about comparing LMMs to these methods and showing that it worked pretty well. But the area that I am aware of that I think these would be very natural fits for would be knowledge graphs. And I'll talk a little bit about what those are if you haven't heard about knowledge graphs. But so these were 4 areas that, just to kind of set the stage as I was thinking through what might guide us through some, examples as to what that would tell us about the standards and the information we would need to provide for these lists to be used in these sort of methods. Okay, so first off, thinking about training or benchmarking data for a new algorithm. And again, I'm not going to be suggesting any definitive things, just thoughts to think about as we go to discussion later. And I welcome comments in the chat or comments at the end. But when you're training or benchmarking a new algorithm, you need this matrix of the true positives and the true negatives, and then also, the positives and the truth data. So that then when you have your predictions, you can compare the positive and negative predictions to the actual truth. So I think it's pretty straightforward that for this, you would need, obviously the truth data, an indicator of which gene is predicted as a true effector gene. But when you start thinking through this, it's not enough now just to have a single gene at the locus. You need to know the negative labels at the locus as well, right? So as Brent mentioned yesterday, it's not enough just to say every gene that's not in the effector gene list is a negative, right? The gene would need to be at a GWAS locus in order to be considered a negative. And then exactly what that means, what is a gene at a GWAS locus? So kind of very clearly annotating what the negative predictions were as well, to the extent that that's possible. And then I think a second thing, here's where having actual, not just rankings or labels or, binary, the gene is or is not, but an actual confidence in the gene's prediction, I think is very, very valuable. Because when you're using input as training data, most algorithms are going to want to weight predictions that have higher confidence more than others, and having a quantitative probability of a prediction like Brent's method, like I was talking about yesterday, would be more valuable for a method that's something that just lists the top gene in the locus or ranks the genes or has scores like, say from the POPs method, that you can order genes at a locus but have no meaning in terms of an actual confidence that that is the effector gene. Okay, so moving forward, so moving beyond just the labels or the outputs is, as input to a method, getting into the features. So, most, I think, methods, it's fair to say, are going to make predictions based on some properties of either the gene or the locus. And I think Eric referred to these as top down or bottom up, and there's others that have been kind of advanced. So it would really be nice, obviously, to make those features available alongside all of the labels. But this gets a lot harder, I think, to define standards it's compare, like, as hard as it is, would be to kind of calibrate a prediction of a gene. It's even harder to standardize all of the potential input features that one might use into a model. Because ideally what you want, right, is to take multiple lists and kind of have all of the features available in the, this, to be able to match them across data sets, to have the values and the units be the same. You really want, like, in other words, like a squared off matrix, because a lot of these methods, missing data is really difficult to deal with. So you want for every prediction, every list ideally, to know like that if you see a feature that is open chromatin in pancreatic islets, that if another list reports something, you can determine that those features can be kind of joined together as opposed to being different. And that gets really difficult, obviously, because how do you, it's very, how do you define like whether open chromatin in this experiment for the pancreas is the same as another. But nonetheless, I think it's at least possible to start using, standard ontologies for like, annotating cell types or tissues and like the type of experiment, and at least coming up with universes of potential features that span the lists that people are using, and kind of fitting features into those. That is a burden on, I think, the creator of the list, but presumably you're putting a lot of energy into creating this list in the first place if you're doing it manually. Or if you're doing a method, then you do have an automated method, then you do actually could standardize these up front. And that would make it much easier for people downstream to make use of these, because it minimizes like the manual munging of different features and mapping them, and kind of greatly aids in data QC of using these as input. One issue here, though, is that a lot of these features are not just like numbers. Like I think the standard way most models run is that you want a numeric or categorical or binary value for a feature, like is or is not an open chromatin. But a lot of these lists that we've taken on, like there's groupings and structure, right? So like the gene features are going to be different than the SNP features, which might be different from other features of the locus. And sometimes those are grouped in certain ways. And, there are ways to represent data like JSON that allows you to impose this substructure, but then that makes it harder to use other tools that like kind of assume matrices. So I think kind of thinking through like, the lists that we have on the portals are great, but they're really just for viewing. Downloading them is a mess. And so thinking about it, are there standard formats that you could download in matrix format if you wanted just the numeric features, or like JSON format if you wanted the substructure, et cetera, et cetera. Okay, moving on to knowledge graphs. So those, just very briefly, knowledge graphs are becoming a very big topic in a lot of areas. And essentially what it works is it's encoding data in these triples, where there's like a subject, and then an object, and then a predicate, which is kind of like describes the link between that, and then some metadata describing that predicate. So here it could be, the subject might be TCF7L2, the object might be diabetes, the predicate might be is effector for, and then the metadata would indicate like all of the, features that we're just talking about that predicted that. I'm showing here one standard that's been developed for these, that's used by the NCATS Biomedical Translator project. It's invested a lot of energy in, and they're trying to advance standards for encoding these knowledge graphs, and this has had some uptake in other NIH initiatives. And so this, I think, is actually, by contrast, an easier lift to get these lists into this format, because to some extent there's a lesser burden on numeric and quantitative consistency, and there's a more flexibility in how you encode these assertions. However, you still need to think about how do we encode, like what are the appropriate predicates for the different predictions. Things sound semantic when you say is candidate versus is effector versus is potential candidate, but those things actually matter when you get into these, data, these like knowledge graphs. And then the provenance, there's a lot of energy that these initiatives do to encode evidence and provenance in terms of the metadata. So all of that would need to be worked out. And then ideally we would, if we're thinking about repositories for these lists, we would have them not just in places you could, browse in the portal, or you could download the features, but also we could contribute these to these knowledge graph efforts, which are much more amenable to these federated, linked data and computation. And some initiatives, like I'm sure the one here that's used by translators called the BioLink model. And smart APIs are a way to register them. So this is I think a whole nother class of users for people. The people working in knowledge graph and reasoning over them are very, very different from the people who are building models and making predictions from genetics. And both of them, I think, could be served by getting the data, albeit in very different formats. Finally, just to touch on, I was initially going to spend a lot of time kind of going through this as a case study, trying to compare these two lists. I think this is for EGFR. And then I just gave up. I didn't have time to do it before today, but I think this example of just simply taking two different effector gene lists by two different authors for EGFR kind of illustrates how difficult it is today to work with these lists. So, just the very first step of matching up loci. So you knew like, okay, this gene, like that was predicted for locus K1 for the first paper is, comparable to this other gene that's at locus G2 in the other one. And so having some, some standards as to what for each predicted effector gene, what was like how do we define a locus and what genes were considered for that, I think is actually non-trivial. I think a lot of efforts I've been in are really struggle to define what a locus is. And you can define it as like a credible set, but then how did you build the credible set and how far did you go along the wings to consider the genes? And did you use LD or did you just use fixed windows? All of that's completely done differently for these. And then obviously if you're comparing two lists, it's like, it's hard to compare a list that has just a single prediction versus one that's ranked versus one that's scored. And sometimes you don't want to be unfair to one list or the other. But I think this just kind of illustrates a lot of the things that I think talking about in the first, just provenance and predictions that, those would go a long way, even to this very basic step of trying to understand consistency across the lists. So then I'm just listing here, these are not my ideas, but just more broadly, AI ML requirements. There's a lot of NIH, supplements that have gone out, and there's a big push, I think, to try to get data more AI ML ready. A lot of these are very high level, and I think the field's still working through exactly what it means to make data AI and ML ready. But essentially, it touches on the features that I just talked through, which is you want the data to be very well structured, so you don't need to spend a lot of time munging it. Ideally, you're cleaning and you're making all the values consistent, and then having standards for the data and metadata. And again, I just want to emphasize metadata for someone browsing on a portal might be different than metadata needed for a knowledge graph constructor who really likes ontologies. But for people who are working in matrices, ontologies are kind of a nightmare. So the first thing I do when I get an ontology is I convert it to not an ontology. So I think that in terms of thinking about we might want to have multiple formats that serve different user groups for that. And then like a lot of it's just about really making sure these data sets are as clean and consistent as possible, even if I think you have to make some compromises by not fully capturing all of the information. Maybe that could be, reserved for one format. And then there's an ML ready that like understandably like maps all pancreas data sets to one feature, knowing that you're going to have some loss in precision, but at least being more consistent and clean. And then finally, just the thing to think about is when we talk about standards, I think it's just worth spelling out that there's lots of things that we could mean by that. So like, what are the required features that every list should have, right? That's what we got to, that's what I mentioned earlier, which is, can we square off the matrix and make sure that every feature is recorded for every list? And then within the features, what are the allowed values? Right? Those are actually separate things. And then there's metadata standards. But as Maria alluded to yesterday, even like the format in which these things are generated, like obviously an image of a table is not going to work. But as I just discussed, like, you might want to format in JSON LD, but you also might want to format in a matrix, and those might have slightly different content. So I think those are important. And then of course, there's a repository. I think ideally, even though that didn't sound like in the survey, that was the main blocker, I do think that having for Hugging Face has been this great repository for all of the LMMs. It would be, I think, very convenient if you could get access to all of these from one location, so that you could just trust that they're all handled the same. Like when I was downloading GWAS data sets recently from the GWAS catalog, it's just very nice that they all have the exact same columns. And, so that once you get your script working on one data set is going to work for the others. And so that's basically it. I mean, like I said, I just wanted to summarize some things to think about. You know, a lot of these I think are straightforward, but I think just the main thing just to flag is really how I think it's an evolving. I don't think it's totally clear what people always mean when they say, I need the data to be AI or ML ready. I think that we should embrace that there are different consumers even within this subspace, and that they might want the data in different formats. And that if there are some, you know, I don't think too cumbersome, hopefully, but some requests or some standards that we require of the submitter, or maybe journals encourage submitters to do, then it might be possible to have pipelines that convert them into various formats that could, I think, really increase the use of these in a variety of efforts that are making use of downstream data. All right, that's all I had. Happy to hear comments or other from, like I said, others who work more closely in this space or who have, you know, other thoughts on it.
So, virtual clapping and online comments.
Thank you.
Thanks, Jason. That was excellent. Questions or comments, reflections from folks?
Yeah, I thought that was a really nice framing of the challenges, and there were definitely some appreciative noises here in the room at EDO. Some of the points, especially about defining a locus and so on. Yeah, really nice introduction to the discussion section, I think. Does anyone have follow-up comments in the room here? Adam?
Maybe just to expand on defining a locus was one thing. I think also defining the sort of unit here of annotation, because there's which genetic variants are going to that, but I think there's also which phenotypes are being looked at, because that might be like, I did a GWAS of coronary disease and I annotated according to this. But then you can go to the other end of things, which is say open targets, where I think, for example, you can put in a variant and it will then annotate that, you know, it finds it's associated with lots of phenotypes and it's giving you a causal gene predictor across many. And of course, there's things in between that. You know, Eric showed nicely yesterday how actually doing that kind of PWAS for a variant and kind of understanding, you know, you may not find it from your complex disease phenotype, but you may be more likely to find the effector gene from. So I think also kind of trying to find what is the set of variants and set of phenotypes that you are trying to ascribe an effector gene to, and having some way of mapping those is going to be kind of important, I think. Because I would want to compare across the traits. I might want to compare that list across coronary disease, as you were saying, like for EGFR, which is not trivial in itself, but then also to compare across almost like, or a set of other related traits as well, and say, actually, well, everyone's saying for obesity traits that this is the causal gene, so maybe for my trait, I should be looking at this one instead. Why does mine give you a different answer for?
Yeah.
And I think that's also slightly non-trivial. I guess maybe more tractable because we have ontologies for phenotypes. But yeah, some combination of those is probably going to be needed to define a sort of, it's almost an association, I guess, a set of variants or a locus versus a phenotype or set of phenotypes.
Yeah.
I'm curious, and maybe this is an exercise we do as we leave this workshop and do some work after this. Would it be valuable for us, Jason, because we work across the space of developing and ingesting these lists, and we also work with folks that consume knowledge graphs? Would it be valuable for us to do the exercise of saying, for what we have for now, for the current 150 lists, how might we convert those into something that is, I don't know, ingestible by a knowledge graph? Could we do that work to help us understand the different types of standards a little bit better?
Yeah, I think that would be good. And like, I think we're involved in this NIDDK project, PancBase, that has a component of it, PancGraph, which is a knowledge graph about knowledge of the human pancreas. And you could imagine this would be like the effector gene list for those traits relative to the pancreas could be a really good example. Something like a broader initiative like Translator, like one of the frustrating things about that project is it's so high level that all of this is boiled down into like the only type of edges that model supports is like gene associated with disease, and associated with doesn't mean genetically associated with. So they just don't have any ability to get into the details here. But for something like if you had a dedicated knowledge graph for browsing then I think it would be really cool because you could see like this gene is connected to this disease, and here's all of like, because the variant is in this credible set that's in this open chromatin, and the open chromatin is in this tissue that's relevant to the disease. So I think that could be a very instructive exercise, actually. And it could be a tractable thing to manually encode some of the lists that we have.
I mean, in fact, for the one you mentioned for PancBase, we actually have a very well curated actually type 1 diabetes lists in the portal right now, where we actually have all the evidence sources, the actual provenance, and even the link to the raw data. That would be a great test case for PanGraph. So we should absolutely follow up on that. I like that. All right, I think we're going to take a break. Does that seem right, Maria, Laura?
Yeah.
That's where we're at next.
Sorry, there's one more question in the room.
Oh, sorry.
Like a really core challenge that's come across in Jason's and Maria's as well. Maria's presentation is that for interoperability, there's like a really, really core problem here in that a lot of these lists have ranking, a lot of them have scoring, a lot of them have categorization. That's like a really basic issue. And in terms of interoperability, what do you think, what can we do about that? This is quite basic, but it seems to be like the elephant in the room. Any thoughts on that in terms of like if you're thinking about your users ingesting this, how are they supposed to start?
So you said core categorization?
So if you think about the list that we see, some of them the output is ranked, some of them the output is scored, and sometimes we have this categorization, you know, causal, possibly causal, all of that stuff. That's a core challenge for interoperability for a stream user. Any thoughts on where to start with that?
So I could suggest too, although I think this would be a great topic for the breakouts, and to some extent, one of the main ultimate goals of what we're trying to do here, right? But I think there's two ways you could start. One is the existing lists. It could be possible to have an initiative to kind of benchmark, come up with benchmarks and ways to like convert post hoc the predictions into some type of like category, right? And you can do that based on, I guess, reapplying some of like, I know there are tools like Benchmarker that have been, there was a paper on that, and some other ways that you could imagine, right? Like validating or post-processing the lists. That's probably not ideal. And I think the best way to do it is forward-looking, and to like encourage people submitting GWAS to produce these lists in the first place. But then second, to come up with some guidelines for how, kind of like Brent was talking yesterday for MR, and if the journals like really encourage this, you could say like one of the things you have to do is come up with some type of numeric score, and it should be a probability, right? That, not that like you have to do it, but that will be like a very valuable thing, or very encouraged in the like when you're submitting a paper. And I think it has to go to something that the people writing these papers start doing more and more. Just like, you know, back in the day when, you know, moving from candidate gene studies to GWAS, I think that this is like a very, I mean, obviously not the same, but it's kind of similar, right? Where you didn't have tons of standards for how the candidate gene studies were done. Everyone kind of had different rationale. And then GWAS came along, and you had like a very consistent input. You had consistent standards, you know, when the p-value hits 5 times 10 to the -8th. And if conversely, you had a set of simple things that, you know, like, okay, it's very encouraged to make predictions, put them on this type of, you know, at minimum apply this method, right? Which we validate, which we recommend, or something. I think you could start to hopefully encourage more consistency in how this is done. I mean, I'm sure that's way harder than I'm making it out to be, and maybe the people involved in journals, or if they're on, could comment on that, or I think Brent had some thoughts on that too. But that's two thoughts I could make. Basically taking the existing, trying to uniformly post-process them, and then second, trying to come up with incentives, carrots or sticks, to encourage people producing these to do to come up with some more, it doesn't need to be complicated, right? Just some simple ways to make them more consistent. I know what others think.
I guess to chime in on that, coming from the rare disease space, other things that I can think of in addition to getting journals involved, funding agencies, also professional organizations. So we've seen that for rare disease, you know, sort of classifying variants is actually not something that has been standard for a long time. And in the last couple of years, the American College of Medical Genetics and Genomics came out with, you know, a set of standards And over time, we've seen that more people are using those standards, and over time, journals have endorsed those standards. Instead, if you want to submit variants, you have to classify in this, standard way. So I guess what else are we doing in this space to, partner with other groups like that?
Yeah, not much at the moment, but that's, I think we need to.
No, but I think that's the type of precedence, right? If it could even, so if you could, the journals could say, if you are submitting a list, right? Like there's these standards that the community has started to develop, and we encourage you, or please, there's a checklist that you've done them this way. Again, it doesn't need to be complicated. Like, to be honest, I think what I'm hoping personally is one of the things that comes out of this workshop is maybe an interested group of people who over the next year or so would say, like, this is really important. We should put out like a draft set of recommendations such that we could, I think Brent mentioned this yesterday for MR, like we could write a paper that says like, these are the standards. You could follow either rare disease. I think that's a great like example. And it could be like, and you could try to balance like what's feasible to do with what's necessary. And it would be, not trivial, but I think could be done with a dedicated group of people who are interested in this problem over the next year or so. And I think it could be really good for the community and good for journals and good for everyone.
Yeah, that's exactly what we're hoping. In fact, I think that's why we invited some of our funding, folks that fund the various portals and activities with our GWAS Catalog, Open Targets, and the portal to this conversation to help enforce those standards from an NIH perspective as well, because they care about this. And I like the idea of professional organizations. I also like the idea of like a lot of us partner with industry, partners. We had Eric and others on the call yesterday who think about, I want to use these. So I think there's a lot of people that can really work with us as we lead this conversation, and hopefully are interested in following us through.
Hopefully can see my slide. Okay. So yes, this is, as Noël said a couple of times, we thought it would be much easier to start discussing the requirements for a standard if we had a kind of model to work with. So yeah, a straw man proposal that we can pick at and poke holes in and change. And I should say that, I'm work for the GWAS Catalog. I know a lot about curation and standardization, but I am not an expert in the field. I'm not really doing this work like you guys are, generating these lists and using them to find out real biological insight. So this is 100% for you guys to poke holes in. That's where we're starting from. So yeah, we wanted to mock up a peg list, or I'd also like to say perhaps we should start referring to it as a peg matrix. And this came up a few times from different people during the workshop, that really we're not talking about a list of genes at this point. We're talking about a matrix of evidence that might in the long term feed into a list of gold standard genes. So we can think about it as a matrix, and it would be suitable for publication in a journal. So for example, presenting as a supplementary table, for submission to a resource, and reuse in a pipeline. And I think Jason also really nicely illustrated those different use cases in the previous presentation, and there might be slightly different needs for those different use cases. So for example, to reuse in a pipeline, you might need additional data that you'd want to necessarily put in a journal. And this is reiterating what Maria presented yesterday. So thinking about the metadata standards, so we started with including the GWAS that the list was derived from. And just to be clear, for the sake of this example, we're focusing here on where somebody has generated a single list from a single GWAS. And we've talked more widely about that there are cases when lists might be generated from different types of evidence, but also that we can think about effector gene lists as being the gold standard list that is derived from several different data sets. And I think we really need the underlying standards for the sort of as a bottom-up approach in order to build those pieces together to make a gold standard list. So for the purposes of this example, we're assuming we start with one GWAS and then we develop an effector gene list from the GWAS. So we need to use standard terminology for the different evidence types. We need to include criteria for significance for each evidence type. And this was a requirement that came up from discussions with open targets, that some users will need to see really easily whether the list contains de novo wet lab evidence or whether it's just purely based on computational processes and using evidence that's already in databases. If there's a prioritization method, the method needs to be detailed. And here again, this is linked to the first point. Here we're thinking about lists that would be generated by a single research group, perhaps in a particular publication. Of course, there are pipelines like open targets that are generating huge amounts of this data. But for the purposes of this example, I'm not really thinking about those pipelines, but the fact that this a standard for individual groups to use must be interoperable with pipeline generated data. So thinking about the standards for the data, so there should be a single matrix containing all the evidence types, so they shouldn't be included in different tables. As Maria mentioned yesterday, it should be a plain text file, not including graphics. The evidence for all genes considered in each locus should be presented. We should be using standard identifiers for genes, and ensemble gene IDs might be preferred over HGNC identifiers there. And it should include the sentinel variant. And again, for the purposes of this example, we haven't considered other types of analysis that don't start from a sentinel variant. But that's something that we can talk about in the discussion. And then the thorny issue of a prioritization or score, and as this question was raised at the end of the last session, can the community agree on a scoring or ranking system, or should we be allowing users to, or rather data generators, to impose their own scoring system? But then the data resource that's ingesting the data had their own score that's imposed on top of that. So then you can see the two together. So the groundwork for this is that UA surveyed all the PEG lists that are currently curated in the knowledge portals. And our goal was to produce a standard format that can incorporate the most common types of evidence that are presented in those lists. And so UA started by categorizing the types of data that were in the lists and looking at the most common ones. And so the most common data elements were things like gene name, variant annotation, and then QTL and tools, fine mapping results, and so on. So that's the process that she went through to develop this proposal. So then I'm going to hand over to you for the next part.
Yeah, so let's combine some of the experience we have from the GWAS catalog, as well as the gene that I learned from Maria and from the same DKP. So we're trying to get some of the proposal as a start point, initial our discussion, get the ball rolling for some more details. So in our proposal currently, we will suggest for each of the gene lists, it's only for a single type or single trait. And a full gene list should be contains two separate files. The first file is metadata, which is a file level description of what the data we have, where the data comes from, what the data looks like, how to interpret the data, and how to analyze the results. And the second one is really the table data, which includes the data from all the evidence for all the genes from one study. So I will give you more details to show what data will look like. So currently in our proposal, we think that metadata should be a file, should be read friendly for both of the humans and the machines. For human will be, if I read this table, read this file, I know what the evidence they have, what the data comes from, how they generate the final score, which makes the research method more transparency. And for the machine to read, say, I got the evidence tab, I got the resources, like I can extract or column information I want to really quickly, really straightforward from this MetaYAML files. So from here, we suggest like have 3 big sections. The first section will be like GWAS data because we think that technically the money comes from the post-GWAS analyze. So tree is really important. We encourage both using the free text tree description as well as the ontology mapping, including like the ontology terms and ontology ID. And the GWAS source is also really important. If that one comes from GWAS Catalog, the GWAS ID is enough. If not, we will also suggest the users submitters to provide sample information about sample ancestry, sample size as well. Since we still have variants, so the genome build is also important to let us know the correct variant. For the evidence part, we found like the source of all the evidence is really important. So no matter which evidence it is, it should tell users where the data comes from. In addition to that, different evidence may need additional information to correctly interpret data, which is also highly encouraged field. The latter part is free text method description to say, or may combine the data or interpret the data in a different way. This one will be more likely to tell users how I combine my evidence, gets the prioritization results, and how you can interpret the final prioritization results. For the data part, it'd be like the table format. It'll be more easier for pipeline to digest. So for the gene part, we suggest to have an symbol ID because it's more consistent with a different platform. The gene symbol will be easier for people to read to see the list. The variant ID is also important. And the middle point will be like the evidence. Since we have lots of different evidence tabs from different samples, so we will encourage like the headers can follow the pattern of the evidence tab underscore with the tissue information, because it can be multiple tissues in one table. And as Laura mentioned, like we think users may have their own score. Maybe this score can better explain their own data, but we still suggest having some universal score or uniform score, which can allow users to compare between the different lists or more easily to integrate among the different lists. So I will show more detail of the list will look like.
Oh, you might need to stop sharing.
Sorry.
Because it will. Sorry, give us 1 sec. There it is.
So here is also the artificial tables, like way the future will be looks like. As I mentioned, like some of the fields I mentioned previously. So we want like all the variants to the single. We want all the genes to the single variant, and we want all the variants in the critical size based on the file mapping results. And as well, sorry. In addition to that, like with for each of the evidence tab, it's not only just in a single column; it can be multiple columns. It's really based on the complexity of the evidence it is. We also support like free text biological evidence based on the biological knowledge and literature review. The literature review results, like following with the PMID number as the support evidence.
Do you want to go back to the
Yes.
presentation?
The right with the sharing the screen.
Sorry, we thought it would be seamless if we shared the presentation. No, that's my other.
No worries, while you're sharing, Mark had a good suggestion that we should, RSIDs should be included. If coding, then consequences can consequences also be included as a question? Just to put a marker down. While you guys are converting.
Yes.
How best to do this? We have about an hour left, which is actually not that bad. I feel like we rushed ourselves a little bit more than we needed to, but I'm hoping that a lot of the discussion that happened within the breakout sessions can come back here and sort of ideally, I mean, just to be very clear, I would love you all to give us very clear guidelines, no pun intended, about what you want us to do next. And by you, I mean a broader you, which is a group of people who eventually are willing to work with us beyond today, to take some of these standards, anoint them in writing, and think about how we can build them into the various resources that are represented here through the GWAS catalog and the knowledge portal and even open targets, to make that into a standard for the field, hopefully lead to a publication, hopefully lead to journals uptaking that concept to enforce those standards, and hopefully a resource that somebody not here could use to make some decisions or support the hypothesis. So thank you, Carl. Go home. I appreciate your discussion. We'll definitely follow up with you. So it's great to talk to you. All right, so why don't we, who wants to go first? Should we have breakout room first go? What do we think?
Sure, Zoom room 1, how about?
All right, Zoom room 1. Who was Zoom room 1?
I was
All
the
right.
leader. So do you just want like some overarching things that we spend a lot of time on, or what would be I don't know that going through every bullet would be best, but you tell me.
Tell me salient points, things that stuck out to you, things that you were like, we didn't get to this, but this is still a problem, something like that.
Okay, so there's a couple of things that came up, and I don't know which question they hit best, so we'll just go with that. We were looking at the example table and some of the fields that were there, and a couple of things that came out is that every study is going to sort of use whatever resources they want in deciding what their effector genes are and how they come up with their evidence. So when you look at columns like PQR, et cetera, that are like expression in this tissue, et cetera, if you do this across all tissues and all data sets, this table is going to get very large and very difficult to use. And so the idea of having some sort of just summary table that's like, this is the Simpson variant, or however we want to define the lead signal, or however we want to represent the signal. This is the genes we consider, this is our decision, however we want to rank that. And then link out to the paper for the actual tables where they give all the details of their evidence, as opposed to having that all in some giant summary that is going to be really hard to parse through. That feels more user friendly. And then for the things you're interested in, you can go chase them down without having, all of these columns. We had a little bit of a discussion about whether or not credible sets belong in this table. Maybe instead we just need some variant that we're using to represent the signal itself. Whether that's the lead variant, whatever that is, as long as it's consistent across how the study itself references that variant, so that you can go back to the paper and make sense of it. That feels more broadly user-friendly and usable than credible sets, where not everyone, agrees with credible sets. They're all kind of generated different ways. And again, you could send someone back to the paper, maybe with a link to their credible set or something for those who are interested. We also talked about when you set up the portal or the system, however, that it's not only going to be useful to query on the gene, but to be able to query on the variant of interest too. Like if I'm doing a GWAS, I'm more likely to type in my lead variant and see what people have already said about it, or links with it or its LD proxies, than I am to go look up every single gene in a region. So finding some way to query on that would be helpful. I guess the other thing we sort of talked about a little bit with versioning is I think that not everyone's going to update their list. So someone like myself and some others in our group, like we generate these effector lists from GWAS. I'm never going back to that particular study and updating that list. I'm going to update that list by doing a new GWAS meta-analysis, and we're going to annotate those results. And so for someone like me, I'm never going to update that list, but there might be others like Open Targets or somebody else who is doing that. So just keep in mind, not everyone's probably going to update prior lists.
Yeah, that came up in our group.
Yeah, I think, I mean, we've got plenty of notes in the slides too. A lot of them are pretty small things, but I think that summarizes a lot of what we spent time talking about.
All right, so that's a question back to you. So these things came up in our group too. I actually like what you said. I'm going to pull back. Updating is something we should touch back upon. And I was wondering if that could be just simply fixed with, if you're going to have another publication on said list, we as a resource would almost be responsible for doing that part, where we would, I think you mentioned this, which is like, oh, look, Cassie submitted a list in 20, blah, blah, blah. And oh, there's another one from the same submitter. Like it's almost like it raises a flag for us to say, do you as submitter, you can even do this, do you want to replace your old lists or keep it for version control? And do we populate that? So we could have that be on the submitter if the submitter has done something historically. That could be a way to just simply circumvent that being sort of scope creep or whatever. That's one option. But I'm curious what other, I think the updating came up with laws in my Zoom room was just like, yep, we have this old list. We've worked with we have a new one. It has all these other things and we're not going to update it. And I was like, ugh. But the same token, I'm like, I get it, you know? So
Yeah.
this is important to actually, particularly if we're going to come online with a resource for the things that have already been produced. Where do we handle updating it? So that's an open question for me, from my perspective. So Laura made a comment that says the relationship between summary table and paper should be the other way around. Laura, do you want to comment on that?
Yeah, I think the way you were describing it, Cassie, you were saying all the information would be in the paper and then you would only share really the summary results. And I've been kind of thinking about it like the other way round. So analogous to the GWAS summary statistics, like you don't put the full genome-wide summary statistics in your paper. You just present the lead loci and maybe maybe just the variant and the p-value in the paper. Yeah. But then you share the summary statistics and then you've got all the data there. And then as resources to ingest those, they've got the choice of how to present them, how to visualize them. You do your own LD analysis and present the top variants, or you can present the whole lot in different ways. So I'm thinking about peg lists in the same way. So that huge unwieldy table, that's what you would share in the resource, but then you've got the option as an author in the paper, you just present the summary data, and so you can find the full results in the knowledge portal or the GWAS Catalog or wherever.
So I see that, but I'm wondering, and I'm not a developer, so maybe this is my naivete of how this goes from my list to actually something online. But if I use GTEx for my EQTL, okay, so I have a column for GTEx with whatever tissues, and then Yakov has a different data source that he's looking at EQTLs, and so he has different ones. How are we going to take that and actually put it into something online where it's like everyone's got data from different sources, I guess, right? And being able to put all that together into something people can actually digest online.
So I think that's where the metadata is really important. So you should have enough detail in the metadata to be able to interpret the numbers that are in the table.
Yeah, I mean, I guess maybe these are things just to think about as we figure out how it's going to look and what we can actually get people to fill in and not. But it was just something that came up as we were thinking about how we would use this list kind of as it is, and whether or not it's too much to try to search through or sift through. So
yeah, no, there's a really good point. I think the way that we harmonize similar types of data from different sources is a real challenge, and I think that will need more focused discussion with people who are doing the work.
Can I make a quick comment?
Yeah,
it sounded to me like what Cassie was suggesting sounded more like the curated top associations in the GWAS catalog. So it's like, this is the summary of the most interesting data from this list or this paper. That's sort of like a UI thing, something I can easily look at on a website and get an idea. Then like the next bit is the full raw data set, which is stored on a repository like the GWAS catalog. And that's maybe not standardized. You're not really worrying that much, maybe at the beginning, about whether that's got all the right columns and if it matches Yakov's data set, for example, in your example. And then the paper would be something again, which is like a story that the author is telling. Yeah, I don't know. There are like there's the big data that you can dump in a repository, and then there's also like a curated thing that you can display in a resource.
Yeah, I think that was a nice way to put it, like the way I think we were thinking of it.
Yeah, okay.
But the display got me thinking. The comments it's like basically almost like you just want to see a checklist, to come to this conclusion that this is the peg from this GWAS. We looked at, we had expression in the right tissue, tick. we had colloque with EQ-Towns for the right cell type tick. We also validated the association experimentally tick, but we don't have evidence for an existing mouse model phenotype. We don't have just I don't know, just a very, a checklist basically of what evidence you have that came to that conclusion that this is your top effector gene for this particular study.
I like that simplicity. I like that. Masika, I see you have your hand up.
Yeah, I like the comparisons to the GWAS catalog, but one of the things I struggle with, both as a user and as a generator of peg lists, I guess, is there's no real sort of clear-cut rubric, right, of how one fit, and this is where we were discussing in our group that it's a little subjective. So I sort of, it's easy enough now with GWASs that we have a threshold and we look above a certain threshold. We know, what the QC is in general, allele frequencies, et cetera. This is where I'm struggling a little. Like if someone uploads and no 10 people are currently, in my mind, doing exactly the same approach to picking their effector gene, how will you go from these crazy tables, whatever someone uploads, to curating that and moving it forward? That's where I think the discussion has to lie, because we may end up with effector genes in your final table that have very different levels of evidence. And as an end user, let me separate the hat of figuring out what an effector gene is. As an end user, if I don't do that part, how will it be transparent to me when I mine these databases that, hey, this evidence is not the same as this evidence, right? I mean, as a community, I feel like that's really important to be transparent about. And that's the difficult part for me. Like uploading a data file, whatever format you want, that's easy. But as a user, making sure that I understand the lack of equality, if you will, in defining an effector list when there's a bunch of genes in there, that's the part I'm struggling with. And I don't know. You guys probably have better thoughts on this, but I wanted to broach it because I think that that's a real struggle for me to think through.
No, it's brilliant. Jason?
Yeah, so a couple of thoughts on this, and this came up, I mean, we'll get to this a little bit in our readout, but the first thing I think it's important to understand that, or not understand, but this is my opinion, that you can't treat these lists like data, they're not data. And these people in the knowledge graph community, they distinguish between like knowledge and data. And like, this is an investigator's like belief into what an inference or conclusion they've drawn from the study they've done. And so there's a different way, I think, to approach the way you present those. It's also, it's important to, so this is now subjective, but so I think instead of trying to come up with like a universal standard for being objective or being quantitative about this, just recognizing that there's some subjectivity, it's still useful and focusing on what are the valuable things to provide alongside that subjectivity. And usually it is very clear rationale for why that inference was drawn. So I think the provenance that gets like having standards for the methods that they use to generate this in various formats, being very like even a high level summary. We talked about like just a category from which that the producer says like, I believe this is strong, weak, whatever. But then there's like a free text form that they justify why that is. And I think that would be valuable. So like, saying like this gene is moderate because it had a co-localized EQTL in adipose. Like, it's almost like the more detail or less detail versus more you put in that would, I think, be valuable information for the consumer of the list. And so I guess my reaction to this is just that we have to be realistic about what can and cannot be done here, and at least trying to provide clarity and the reason for why the inference was made will hopefully address some of those things. It's just not going to be perfect, but you know what I mean? And So go ahead, yeah.
Yeah, no, I think that that's an excellent point. I recognize that. I guess the transparency around it and how we make that really clear is key. I like the idea of the depositor having their 3 categories or 4 categories, but I think providing guidance on how one does that would be really important, because I'm guessing just so that even as a user, to say, okay, what if I want to go after the things that have the moderate evidence? I have some sort of understanding that the definition of moderate was somewhat similar. Otherwise,
Yeah.
being able to pick that as a drop down won't be useful. Does that having some guidance? It's not, as you said, a standard, but saying, listen, if you have at least this evidence, you could probably label it this way.
Yeah, you can kind of bin them based on.
Yeah, otherwise we're going to have to go after a text field to figure out how did you define moderate versus how did someone else define moderate, right?
So kind of like a cat. Yeah. So here's categories. Here are some examples. There's like some guidelines for it. Another interesting idea, I forget who in our group, I think it may have been Maria who came up with it, was that you can be still qualitative but unbiased if you have like a body of people who are not the submitter also review and label, like, so you could have like I think this was on the template sheet, which was like submitter's confidence, where you have the guidelines that we just talked about. But then it's like there's some committee of people, like I think similar to like the rare disease space, who might say like, we reviewed the evidence here, and like just like they have the Gen CC has like different categories for the genes, we can say like, this is past followed. So these lists are not so commonly coming out that wouldn't be possible to do. There's a lot of thumbs up on somebody. Well, that would be some work. I bet you could get like a committee of people who would be interested to maybe once a quarter review like the new lists and determine, what they are. And so that could be another way to be more unbiased and more uniform, even if you're still subjective.
I love that. That came up in our group too. It's like a ranking by the submitter, and like a probability that we kind of had them to select based on a set of criteria, and something that we do as resources do to sort of summarize that with a confidence or review, and have that be something that the community is involved in, not just us as free-willing resources. So something that has the curation component from a community. That came up in our Zoom room too.
The other thing, the other thing I'll add is, and I don't know the best way to implement this, but something to consider is the ancestry of both the GWAS and the data sets that are being used, because there's a lot of, we even still, our GWAS and our extra data are all Eurocentric, although we're trying to get better. And so finding some way of indicating that would be useful and important, and not hide it in the paper itself that you have to go cite, but actually putting it up front.
Exactly. So that's definitely a criteria. I see UA has a hand up.
Yeah, it was just to follow up on the conversation around getting the community input as well and the evaluation. In Panlab, we created basically a crowdsourcing tool so that you could ask experts in that field, what they thought of the gene, and ask them to provide sort of the underlying evidence for that. So that you could also utilize something like that, crowdsourcing tools.
Yep. We also thought of as a resource, we could add things like if we get an effector list from somebody and it comes up in a Mendelian disorder, we could actually add that as like automated evidence updating, or mouse knockout or something, and which we'd have to come up with criteria for that. But that could be something we could add just to bolster right away the gene's evidence as a resource in addition to the crowdsourcing. I see Maria has her hands up.
I just want to say that we want to make sure we can hear from all the groups, so we should probably get a readout from the other groups.
I think we're doing that across the groups. Like some of the things already came up, so I think that's a good idea. So why don't we anything else, Cassie? All right.
Nope.
Zoom room 2.
No, I think that was yours.
Oh, that was me. Beautiful. Okay. Actually, So I had a great room. I had a combination of folks who've been submitting lists, who are generating lists, who are consuming lists. I had a great group of people. I also had a group of people that were in person as well. So it was like a little bit more. So it was very, very fun room. So anyone who's on that chat, you know, that wanted to pipe up and they didn't represent well, please speak up. So I think to cover some of the things that came up in earlier sessions, a lot of the things that you guys talked about were the same things. So we talked a little bit about insisting on the Sentinel variant. That was a key. Credible said, yeah, but if you have them, tell us where they are, what cell types, what tissues, and that could be in another field that is one text field in addition, so it's not this huge thing. That definitely came up. What other things came up? Ideas of other evidence sources. So this was, again, evidence for rare disease, mouse knockout, other human phenotypes, things that could be human curated and added. That was critical and very useful. Transcript and specificity of the target tissue. How do we handle that? That came up. I'm just raising issues just to kind of whet people's appetite. Defining formats for updating. I think that was, we already talked about that. What other things that haven't come up? So we like the idea of scoring. That came up as well. One of the things that I think our group said was, hey, I'm not going to update my list again, but I want you to know that I did update my list if I'm going to submit it. So by doing that, we need to have some sort of submission or accession or version control in the lists, even if we allow the multiple lists from the same submitter to populate and be accessible to the community. So we want to make sure that that's controlled and there's some sort of like checkbox that says like, this is a new version of a list that was done earlier. So that must be done. Very simple stuff, but very useful. What other things came up? Please automate things for me was another comment. Like, if you could say like, I'm going to give you the Sentinel variant, can you map it to the nearest gene for me in the same version of the genome? So do some things for them that would make it easier for them as submitters to do their job. So I think that was a theme that we heard. Other themes that came up that I'm trying to like pick and choose from things that didn't already come up. I think actually a lot of it was covered, but I would say that what I heard from that was there were a lot of people who are interested in working with us beyond today. So that also was a nice theme that came up, that people are really invested in doing this. And I think there's a combination of folks who are willing to help us build standards, but also contribute their lists in the context of helping us build more standards. So you have that going on too. So I thought that was kind of a nice takeaway that I heard from the session I was in. That's just my readout.
Awesome. I think then we can hand it off to Jason for Zoom. Can you hear us?
Yep.
Yeah. Okay, so in our Zoom room, we had we were heavy on people with curation expert. We had journal representation and a couple of consumers, I think, mostly. So I think the three topics we touched on, one was just whether we believe that it's actually time to work on this problem. That was kind of my suggestion. Just, I think it's there is like inherent like tension, which is it too soon to insist upon standards, or have we reached that point yet? I think we agreed that it's probably we don't want to treat this problem like the GWAS, like standards, like maybe at the point of Mendelian randomization, like Brent mentioned yesterday, or GWAS submission, because those are like the primary outputs of a study. This is more still rapidly under development. A lot of it's subjective, but that it was still worth I think we settled upon the guidelines, like I just mentioned earlier. And then we talked a little bit about the data versus knowledge. So I already covered that. And then we spent some time on the universal, to the extent to which we could have a universal ranking scheme. And again, I think I already mentioned the output of that, which was just a couple minutes ago, about basically embracing that it's going to be subjective. So trying to come up with guidelines for how both submitters and maybe review bodies could help standardize those. But I guess and just emphasizing that the focus I don't want to hopefully I'm speaking for the group and not just myself, but that the biggest value here is on guidelines for making it clear how the list was produced, and having standard formats for it seems to be the biggest low-hanging fruit here. Even if we're not quite ready yet to solve harder problems like making sure the exact, harmonizing across different input like input data sources, making that might be hard to do immediately, but there could be some low-hanging fruit just in terms of, if you're going to submit it, here's the format, here's like some guidelines, and trying to be more transparent about how they were generated, I think were the main themes that came out. Correct me if anyone, I missed something, someone please chime up, but that was basically it from us.
Awesome. All right, I guess we can hand it over to EVI.
Yeah, I think a lot of what Jason was just covering, we kind of agreed with that for this stage that trying to create a universal score is, if that is kind of for the future, but what we really need to, so taking the questions as a kind of a guide in terms of metadata, in terms of the universal score thing, it's early for that, but what we really need to mandate is that people tell us what evidence they use to create whatever their own scoring system produced. So they can submit whatever they want, they can rank it, score it, classify it, whatever, but they need to submit some kind of, say what evidence they used. So that metadata needs to be categorized in categories, and as the straw man includes something like those, but it needs to be flexible for the future. So there should be like an other kind of evidence that people can include. But a minimum categorization and provenance of the evidence types that they use to reach their conclusion is kind of the key thing there. Other metadata questions, credible sets could be encouraged. There's a couple of other minor things that could be included in the metadata. Some way to distinguish between new experimental data versus just data that has been accessed, but that could be clear from the provenance. What else? Oh yeah, in terms of again, I'm trying to focus on things that haven't been covered already. In terms of the format, if we do provide a format with field headers and data types, it should be clear that they should only be populated by information that the user actually used to create the list. Not, they didn't just retroactively go up, look something up in IMPC. But if we do create a format, we should also acknowledge that that is a useful outreach tool, that it can tell people like, these are the kind of things that you can look at, this is the kind of information you need to be including. So I think that's an additional point. What else? Yeah, I think our main thing is that the user should be able to supply anything that they want, but needs to supply underlying evidence and the provenance and classification of that evidence type. And then lots of things can be done with those minimal pieces of information. Am I missing anything?
Beautiful. Reactions, thoughts before we go to the next breakout? Okay. What's next, Mackenzie? Who's next?
Us at the road.
All right.
Yeah.
Go for it.
So here we have some methods, some data generators, some data curators, and some analytical methods generators. And so we basically, I think our group agreed with everything that's been said so far. We talked first about sort of quality standards for the list. Where do we get the lists come from? Is it okay to have unpublished lists, or do we need to have peer review as a quality criteria? And what if a publication is retracted? Who's responsible for QC? These are important points. And let's see, we talked about not making the barrier too high. As Cassie said, if we make the barriers too high to submitting these lists, people are not going to submit them. So unless there are requirements from journals and so forth, then that would be very helpful. But we don't want to make the barriers too high. A really interesting point that no one else has brought up, because I think we have the only mouse scientist in this group, is people are doing these lists for mouse and rat also, and those could be extremely interesting for humans and vice versa, and the same standards could apply to them. So that is something we should think about. Would be great to have connections between mouse and rat and human in this area as well as in other areas that we're working on. And maybe other model organisms, I don't know, zebrafish. Let's see what else. Talked about a little bit about how to define a gold standard gene, which is difficult. Probably Mendelian disease, exome sequence analysis, and successful drug targets, mouse knockouts. All of these, if they're carefully curated, are really good sources of high quality evidence to say this is a gold standard gene. Let's see. We agree with all the metadata requirements, but one that no one has mentioned, I think, is that it'd be really nice if the authors had to submit a free text abstract or paragraph describing what they did. This would be really, really useful for just the more casual consumer of the list to just get a quick idea of what was done, and it would save curators from doing that, which is quite time consuming. Let's see. Julie had the idea of making a tool to help people, and as someone else already mentioned something like this. If we could have make it easier for authors, if they could enter a list of genes or loci, and then some automated analysis would be done automatically for them, like running VEP, nearest gene analysis, and whatever else can be done just automatically in a standard way. That would be very helpful as a starting point for creating these lists. I think, did I miss anything else? I think that those were some of our major points.
Yeah, let me just expand on what Maria said. I think there's, I'm certainly the only mouse guy in this room. I don't know about all the virtual rooms, but I think there's tremendous synergism possible by considering peg lists from model organisms and integrating that with human GWAS that really hasn't been fully explored and harnessed yet. And we're doing exactly what was been described here, generating matrices with as many different columns for features as we can dream up, and a gene where, and I think we said it correctly, a gene where the tick mark is as many boxes as can occur, that gene flows to the top. So we're doing that sort of curation to identify candidate vector genes to explain physiological QTL right now. And I think there's real potential in accepting, and again, peer-reviewed peg lists from model organisms into this platform. And I selfishly raised that as a possibility.
That's okay, you're allowed. That's awesome. I think it's a great idea.
And we also talked about not becoming the police officers of enforcement, and letting the journals do that. And if it's a peer-reviewed journal, then that presumably provides some safeguards that it's high quality. And we know sometimes those are retracted for one reason or another. But put the onus on the authors and the editors of the journals to make sure it passes a QC. Because if you don't do that, I think there's going to be not high quality data that gets through, and we don't want that.
If I can just add one thing. So I totally agree that putting that on the authors is the only way to go. I think that you have to, when modulating the behavior of a group of humans, you have to think about incentive structures. So what are the incentive structures for those humans to do that? So these are academics doing academic research, and they require publications. So if you put that QC step at the entry to publication, then it generally gets done. By having journal editors do it, it means that they're the police officers and you're not, which is much better. And it also allows for a barrier to admission to the knowledge portal that's much more robust than peer review, because peer review, frankly, is. And so I think that as I pointed out yesterday, lots of communities have already done this. It can be achieved by just putting in place a guideline for publication that is accepted by journal editors. This takes time. So Jason pointed out that the community might not be ready for this, but it will take years to do. In my experience, like probably 2 years to get it out. So the getting people like I see Kyle on the call involved in formulating that process, getting a community of motivated people to write those guidelines, stick to them, describe them, and then make them part of the ethos of the community and barrier to admission to publication. They'll solve a lot of the problems that you're trying to solve. It's an expensive time cost, but generally improves. That's all
I think, Brent, that was a perfect sort of summation of some of the things that we're hoping to use this workshop as to get to the next step. So yes, I think so. It sounds like we have our work cut out for us, but I love your idea of the strobe guidelines from MR, applying that to this field. I also love the idea of trying to have an anointed group of people to take a next step with us. So I was thinking about that. Do we have another breakout room to go through, or do we go through everybody?
Worth it. Beautiful.
So we have 20 minutes left, which is actually what we wanted. That's crazy. How do we do that? So with that, I think it's sort of a lot to pull in and a lot to summarize. And I think we're not going to try and summarize our conversation. We're going to do that. Maria and Laura and I and others are going to work to do that, to synthesize that. But I think it sounds like, from what I'm hearing, we are at a stage where it's time to go to probably a working group of people who want to figure out how to make these standards into real things, to summarize what we've talked about here, to consider things like other resources that have done as well, like GenCC, reaching out to them, consider do some research on the strobe guidelines, talk to journals. I'm sort of pulling in different things we should be doing just to give you guys a sense. And then also maybe do some benchmarking of some of the current, my Wi-Fi is unstable, so hopefully people can hear me.
You're good.
Do some benchmarking current effector lists that are already accessible on our resources, maybe working with GWAS Catalog to sort of consider an exercise of back populating the ones that are on the knowledge portal into the GWAS Catalog sort of straw man and seeing how we do. And I know that sounds funny, but I love it if there's postdocs or people in people's labs that are interested in working with us. We'd love to hear from you and have you like knock on our door, virtually that is, to say, I'd like to work on this, because there is room for that, because those things would lead to publications and part of this white paper. So I'd like to invite other people who are interested in working on this problem with us from the different levels that I'm sort of laying out. Another thing I heard that I thought was interesting is I heard some people like, I'm about to generate a list. You are the people who I want to be talking to like right now, not because we're going to make you go enforce all these standards, but this will help us create these benchmarks. What can we do in the process of having you upload? Could we involve GWAS Catalog? Could we involve the knowledge portal all the way through the process as a means to say, hey, this publication went through this process as part of, the overall community working through it. But you can even have that as part of your work. We'll make it easier for you. But I'm just putting that out there to folks who might be doing that in the near term. Those are just some thoughts I have. Maria, Laura, others?
Yeah, I can step in. I think that was a really good summary, Noël. Thank you. I love all those ideas. Certainly trying to, come up with some recommendations and seeing how existing peg lists can be coerced into those formats. And also doing some outreach with the community and finding out whether this is usable, is this easy to generate, thinking about the tools as came up a few times, like, do we actually need people to be mapping their own genes? Can we do that? Much of that process can we do for them? How much controlled vocabulary can we come up with versus allowing people to write free text? I think these are all really interesting challenges that we can focus up, with a smaller group potentially going forward. And then I think, as you said, this is a very long-term plan and would take several years. But I really think this is laying the foundations to get these kind of this this data and this information in a standard format, so that, you know, in a few years down the line, we can come up with some kind of universal ranking system, and go to that point where we have got a gold standard effector genes for common diseases. I think that's the ultimate goal.
Definitely. Maria? You have any
I don't
comments?
think I have anything else to add. This all sounds great.
So I think we actually have, what other comments and things we tackle together in our remaining minutes? Well, I think I'm going to do some programming notes. So what we're going to do is, because you all spent your time with us in the last 2 days, I hope we have all your mailing lists and email addresses, because, you know, you are part of the beginning of this community. So hopefully there'll be follow-ups from this. But one will be all the documents, all the presentations will be accessible through the Google Drive. We'll make the recordings available as well. Those will be publicly accessible as well. That's one step. If you do want to participate in said working group, or whatever we're going to call it, a writing team or a community coalition going forward, please reach out to Maria or Laura or me, or Mackenzie, or anybody that you've gotten to know. We'll all find each other somehow. To contact us to say you want to work with us, because that's important that we do find you if you want to. Other things that we'll be doing in the near future. So one thing we're hoping, just in terms of just like making sure that we hold our feet to the fire, at ASHG, which is a great opportunity to share sort of reach out, we have an ancillary session, which is like a session that's not like a platform talk, but it's a session that it's in the program. And it's an opportunity for us to talk about things in our community that we've done together. So we're going to have a readout of what we all talked about here at that ancillary session. So if you're going to ASHG, or if you know somebody that we should involve or have a conversation at ASHG, if you're going to Denver, that's another place for us to reach out. We'll contact you about all this, so you know this. You don't have to write all this down right now. But Maria is planning to summarize what we've learned from this workshop and some of the salient points, and make a charge to the community beyond this particular group, because we're not going to reach everyone first time around, as to how we might advance this initiative. Particularly because at ASHG, you'll also get the rare disease community attending there in some cases. So that's a great opportunity for that. We'll have a webinar. Our public webinar for the knowledge portals is also another setting with where we'll echo what we've learned together here. We're hoping GWAS Catalog and Open Targets will echo it as well through other channels, because they have great forums for that as well. So that's one thing for you all to know. Other notes or comments that people have that might have come up as I started talking?
Yeah, I have one thing I wanted to raise, actually, Noel, that came up in our group, and this is really sort of getting into the details, but it would be useful to have a flag to indicate when people are including experimental data that they've generated themselves versus data that they've looked up in a database. But so, for example, you know, I generated a knockout mouse versus I looked up the gene in IMPC. And that seems like a really simple flag to have, but that was very wordy, what I just said. I don't know if there's a simple way of distinguishing those two things. So if anyone has a good suggestion as to what we should call that, I'd be happy to hear it.
Submitter generated evidence.
We have
That's still quite a mouthful.
Yeah. Submitter generated.
Yes.
What would the other one be? Database generated evidence.
Yeah, sort of publicly available. Yeah.
How about internal or external?
Oh yeah.
Yeah.
That's simpler.
Yeah.
Yeah.
I like that. I like that. Yeah, that's a great one to pull out. And also there's also, that brings to, like Laws was mentioning in ours, saying they build their list off of a lot of data that's not publicly available yet. Like because of, you know, the fact that it's in their consortia, people are publishing on their experimental model and yada, yada, but they're generating lists based off of it. So that's another thing, which is external, internal, or pre-publication, publication. particularly for those evidence sources that are underlying, like the real perturbational data, like you were saying, Laura, those things tend to be not always published or protected access.
And also to follow up on what Maria was saying about having a kind of abstract summary of the data that's in the table. I think it's ClinGen have a framework for creating a sentence for the evidence, so that, but there's a real structure to it. So I wonder if we could do something similar to have an overall summary of that, like a human readable summary. If that's something that people want, but to kind of structure it and help people write that, so they don't, you know, just paste the entire text of their paper in there.
Yeah.
Abstract.
Which we do sometimes see in the README files in the GWAS Catalog.
Yeah.
We could probably ask ChatGPT.
Yes, I was thinking actually that could be something that LLMs could help with
a structure.
Yeah.
But you're thinking a sentence for how you're using the evidence rather than like for each gene.
Yes, I think
It's not like Eric was saying where he created, you know, for each gene a sentence saying why it's the causal gene, but you're talking about more about the evidence.
Yeah, an overall summary of the evidence. Yeah, your approach. Yeah, that's the best.
I think the portal had something like that. Was it a paragraph rather than a sentence that we had to kind of pull out to put on a page on the portal?
Yeah, we had that. We have also the figure. The figure is the most useful, to be honest with you. That like that figure that describes what you did is also like a graphical abstract. I know that sounds funny, but that was that resonates to everybody. I know that's not scannable, but if you have that, landing that on the actual like list or matrix, you know, place would be valuable for the user to consume. So I was also thinking as we're talking, you know, we're talking about writing white papers and having readouts and working groups. I think maybe to keep momentum going, we make even, you know, this, I'm just not sure what people's appetite for another workshop of this length, but there could be another workshop in said time that has a slightly different end goal, which is like, we digested this, we did some benchmarking, we've gotten some more lists in, here's actually a more formulated straw man, you know, he's got some clothes on now, and sharing that back with the community as maybe a next goalpost for us to get to in like 3 months or 6 months time, right?
Yeah, sounds like a great plan.
All right, I'm cognizant of the fact that we have a lot of international participants who are probably tired from the end of the day. We've had 2 really great days. I really enjoyed this myself. It went so smoothly. So Mackenzie, Maria, Laura, Yue, awesome in so many ways, just really awesome, like just technically, but also scientifically. I think this was really enjoyable. I appreciated everybody coming on site, for those that did. Folks that took time out of their day to join virtually. That's always really awesome. I know that means that people care. I thought that we had a great representation across industry, private, public settings, academic, non-academic. Folks have been contributing, consumers. So I actually really grateful to see that. I think we need to target more consumers in the future. I think that would be fun to really get some people who maybe some naysayers, like I don't really care about these. Tell me why I should. And sort of like figure out how to crowdsource those people. But first of all, I want to thank you all for attending. I want to thank Maria and Laura particularly, and Mackenzie. Any other closing remarks from anybody at any of the sites?
I've had a really good time too. It's been really interesting. Really enjoyed all the presentations. Thank you everyone for your input. We had some really good conversations here in the room at EBI as well. Yeah, I'm looking forward to carrying on with them. Anyone from
thank everyone too. I just want to thank everyone too for making the trip here and virtually and in person. So it's been great.
Yeah, thank you. Yeah, anyone from Open Targets want to?
Just to say thank you, everyone,
Yeah.
for organising. And yeah, we're looking forward to kind of working with everyone moving forward. Yeah.
Great, thank you.
All right, we're going to give people some extra minutes, and I want to say thank you again, and take care, and we will hear from you. You will hear from us again. Take care.
Thanks, everyone.
Thank you.
Bye.